This time we're going to start mid-level and move up into shallower water. Hopefully we'll see something different from what we've seen in previous dives. This is a dive as part of the climate connections at the IC interface cruise on board the Falcor 2 from Schmidt Ocean Institute, and we have live stream from the ROV SuBastian. My name is Dr. Matthew Lee. I'm an investigator at the Centro IMAR, Universidad de los Lagos in Puerto Montt, and I'm part of the Chilean benthic group. So I have with me Dr. Amerigo Montiel and Dr. Christian Aldea, who are from the Universidad Magallanes. We also work with Dr. Eulogio Soto at the University of Valparaiso. Also on board with us we have two students, Benjamin Aguirre from the University of Valparaiso and Leslie Novoa from the Universidad Magallanes. And our group in particular is interested in the fauna that lives in the soft sediments. So I work with meiofauna, my colleagues work with larger infauna, the polychaete worms, etc. And Christian is interested in the epibenthic, the larger organisms that you can see on the screen now, like those holothuroidian sea pigs that are quite abundant in this part. Buenos días a todos. Es 10 de la mañana, hora local. Eso es otra inmersión enfrente de la plataforma de hielo de Jorge 6. Eso es una inmersión en que parte del crucero de Climate Connections at the Sea Ice Interface. Estamos a bordo del buque de investigación Falcor 2, que parte o pertenece del Schmidt Ocean Institute. Y ahora se puede ver en vivo desde el fondo imágenes del ROV o del robot submarino SuBastian. Mi nombre es Matthew Lee, soy investigador del Centro IMAR, Universidad de los Lagos en Puerto Montt, y soy parte del grupo de investigadores estudiando los ventos en Chile y Antártica, obviamente. Tengo conmigo doctor Amerigo Montiel y doctor Christian Aldea, que son de la Universidad Magallanes. También trabajamos con doctor Eulogio Soto, que está de la Universidad de Valparaiso. Y también a bordo con nosotros tenemos dos estudiantes, Benjamin Aguirre, que es de la Universidad de Valparaiso, y Leslie Novoa, que es de la Universidad de Magallanes. Nuestro grupo es estudiar los organismos que viven en el fondo blando. Personalmente estoy interesado en los meiofauna. Mi colega Américo trabaja con los poliquetos y otros gusanos que viven en el fondo. Y Christian tiene interés en los organismos más grandes, como los holothuroidians o chanchitos marinos que se puede ver en las imágenes ahora.
Check. 785, 786.
786. I should also mention that these cruises are part of the Challenger 150 program, which is a group of deep sea biologists and research scientists with support from the United Nations, and the idea is to coordinate research so that we can fill in the gaps in our knowledge of the deep sea environment. También debo mencionar que estamos parte de la serie de cruceros que son parte de la Challenger 150. Es una agrupación de investigadores de mar profundo apoyado por las Naciones Unidas. La idea es coordinar nuestras investigaciones para llenar los huecos que tenemos en nuestro conocimiento de mar profundo. as you can see in the images that we have here, the deep sea sediments are dominated by echinoderms, the spiny skinned creatures. So the most abundant appear to be the holothuroidians, the sea pigs, sea cucumbers. There's at least 3 species here. We've also got quite a lot of urchins, sea urchins, ophiuroids, brittle stars. Also seen some crinoids, sea lilies. There's also some worms. I've seen some acorn worms, hemichordates. I've also seen some echiurians, the spoon worms. And there's been a few ice fish as well knocking around, and there's also some scallops. They're almost transparent if you see them. Como puede observar, los sedimentos de mar profundo típicamente son dominados por los equinodermos, con piel con espina. Y lo más abundante que se puede ver aquí son los holothuroidios, los pepinos de mar, o en este caso chanchitos de mar. Hay por lo menos 3 especies. También hay rizos, hay ophiuroidios. También he visto crinoides. También hay algunos tipos de gusanos como la hemicodata y la echiuria, el gusano de cuchara. También hay ostiones y algunos peces de hielo. At the moment we are doing some video transects, which is basically we cruise along not too fast, not too high above the sediment, and these videos are recorded and later will be analysed by colleagues to identify the species and the abundance, their distributions. En este momento estamos haciendo video transectos. Ese video son grabados y más tarde algunos colegas van a analizarlo para determinar la diversidad de especies y su abundancia y distribuciones. ¿Ya? Entonces andamos no tan rápido, como con una altura de como 1 metro por sobre la superficie de sedimento. ¿Ya? Vamos a hacer eso para los próximos 200-300 metros.
Also, a super quick shout out to the homeschooling students tuning in. This is very exciting. Would you like to let us know where you're tuning in from? Be happy to give you a shout out. And also let us know if you have any specific questions. We'd love to answer them. And also in the picture-in-picture inset, you're seeing a little map of where we're going right now. So you can see the little yellow box, that's our ROV SuBastian, our remotely operated vehicle, accompanied by our ship Falcor, of course.
these species here. So
Ready? Are you one?
Yeah, we'll do
flat
it.
area or?
this area will be fine, and there's quite a lot of them because we need 3. That's a group there. We're going to collect them with the scoop, please.
No problem. I think the scoop's in the rock box, right?
Yeah, usually. Three flying by.
It's all the way out, huh?
Yeah.
Okay, so at the moment we're going to collect some sea pigs and some sediment. This is for an analysis of the trophic links between in the food web on the seafloor. Entonces ahora vamos a tomar muestras de como 3 chanchitos marinos y son algo sedimento. Es para analizar las redes tróficas que hay en los fondos marinos.
It's too ragged.
Yeah.
It's snowing outside.
There's 2 over there.
Yeah, there's 2. These 2.
Zoom.
channel, right?
Yeah, just jump over there. There's a bunch over here as well. So they're pretty much everywhere.
It is so light.
Yeah.
Bounce right out of there, you know?
Yeah. I'll roll away underneath the rock.
The big one here.
Yeah. Yeah, so I guess we'll stick it in ply box to the
To be.
2B.
Did I extend some of your portrait?
Or I just want to make sure we see it go in, you know?
Maybe the words have been your.
Well, both are empty, so.
Yeah.
So you go in?
Yeah.
Is it an A?
It's now an A, right?
Yeah, I think so. Yeah. So. So, 2 more like that, please.
And Transatlantic question in the chat. The ROV is operated on the surface, so we are all together sitting in a computer room. So we have lots of screens here, and the ROV pilots are sitting in the front, gathering all the samples for us.
About the rocks, submarine outcrops of rocks have been observed that correspond to meta-sedimentary metamorphic rocks that could have a Jurassic age, that is, around 150 million years old, but that will have to be verified with geochemical methods.
their defense mechanism. It's just inflate all the way.
Too fat to attack.
Are they like rigid when you bring them back up, or
Yeah,
no?
they're quite little balloons.
Huh. Interesting.
Jelly balloons, yeah.
Yeah, and just to follow up on the ROV question, it is attached to the ship via what we call an umbilical, which is basically a very thick cable, and it's got electricity going through it as well as fiber optic data. So that's how you're able to see the video right now.
Are we on a slope?
Maybe a little bit, it's hard to say.
So pack it from the other side. It's in the roll away.
Yeah, it's just if I do it the other way, I can't really see, you know? But try if you want. These freaking guys. Should we get just suction?
The preference is to scoop, but plus I think
Be a lot funnier.
they get stuck in the tube. I think. I want to know that exploding incident.
You know what?
Remember that the nudie ramp we tried to on the river cruise that exploded when it started sucking it?
Oh, yeah, what happened?
The chunks.
Got like blenderized, right?
Yeah.
Watching out.
Alright.
One more.
Did it go in A or B?
B, I think.
I think that one would be.
You could confirm when afterwards. You could confirm where there's a.
Yeah.
You don't want to mark it there. So
No, no. I mean, genuinely not space.
There's another one right there.
Yeah, there's two right below you. Nah, fish just didn't move. It's not interesting, is it? assumed anyway. Okay, so now I need to jump a few meters to understand a bit of sediment and then collect 3 push cores.
You can close that box too. You guys can float out of them. Drop those back to itself.
Continuing.
Now we need to collect push cores.
Push cores.
Yeah,
Right here.
right here. Need to be associated with the seabed. So we're just going to collect 3.
Are these more bio or more geo or
These are bio ones, so numbers 1, 2, and 4.
1, 2, and 4?
1, 2, and 4.
Keep it along. Which one?
No, this is the push core. It's like that. Sí, gracias.
Ay, dejó una nota.
So okay, now we're going to collect 3 push cores also. These are to be associated with the sea pigs that we just collected. This is so we can get an analysis of the organic material that's in the sediment. The rest of the push cores will be collected later. Yeah.
one is the long one, or
No,
not
we'll
sure?
do number 4. The long ones are for the geologists.
Number 1, 2, and 4, you said?
He visto on
Which one? Pencil?
So you want to do 1, 2, and 4, right?
Yeah, 4. Yeah, the long ones we leave for the geologists.
Okay. 1, 2, and 4.
Thank you very much. Right, we get back to video transecting now.
video.
Yeah.
Pretty edge RV, new waypoint.
Updated waypoint.
a really cool question in the chat, and they asked if, like, for all the scientists here in the room, what's the coolest thing you've ever seen in your career? Like, what's your favorite animal? What's your favorite thing that you've seen in the deep sea? Matt?
I don't play favourites, but I like marine tardigrades. Yeah, they're quite cool. So.
Have you seen a cool one that you really like?
Well, I found a new species on the Seamount cruise.
Yes.
Yeah, there's a photo of it on the International Association of Meiobenthologists website if you look for it. But yeah, all covered in spines.
So I imagine it's a pretty cool thing to describe a new species, isn't it?
When I eventually get around to it, yeah, it will be.
I actually have a bucket list in my phone.
Oh, you do?
Yeah.
What's on it?
It's a secret. No. I mean, some of the things I've already seen, like the phantom jelly from last year.
Oh, yeah, that's also on my
That was
wish
definitely
list.
on my bucket list. I still have some things I would like to see on my bucket list, like a whale fall.
Oh, yeah.
Yeah, and amongst some other things, but yeah, I think that would be the biggest thing, would be like to see a whale fall. I haven't seen one of those
I
yet.
actually agree with that. I'd love to see wave fall and like
Yeah.
all the fauna on it.
It's such an interesting ecosystem.
Yeah.
Yeah.
I do an entire class on it in my ecology course.
Really? Oh, that's so cool.
It's a good example of succession.
Yeah.
Yeah, we actually had a whole lecture about wave fall ecosystem in our university, which was pretty cool.
Yeah, they're also a bit unpredictable, so you have to be lucky, unless you've seen one, a
It should
specific
be-
one before.
Should be quite frequent, the calculation. I reckon if you went actually looking for one, you'd probably find it.
How would we go looking for one?
Well, for example, a few years ago there was a mass stranding in the Gulf of Penas, where we were on the Canyons cruise. I don't reckon all of them made it to the beach. I think likely some of them sank
Yeah.
in the boat. And probably if the weather there is notoriously bad. It was when like
That makes
6
sense.
meters swells.
That makes sense. Is there still anything left on your guys' bucket list?
I'm fascinated by everything. So that big polychaete we saw the other day was good, cool. Never seen one around here.
What did it look like on the surface when you?
Well, all the scales came off it.
Oh no.
But he still looked very beautiful.
Yeah.
It was just a little bit sad that all of the scales
Well, they all-
fell off, but
they always come off.
but it was
Yeah.
big. Like, you don't really get to see the size of the individual, but once it's
Yeah.
back on deck, you just like see it, and it was a big one.
Yeah, it is hard to pick a favorite ecosystem because they're all so different. Like methane seeps are so different than hydrothermal vents, and
Yeah.
yeah,
Updated
it's-
waypoint.
might have bucket list items for each one of those things.
Oh, that's so cool.
You know what? Let me actually see in my phone. I might need to update it.
Crinoid's getting ready to swim.
I still really want to see a barrel eye fish.
A barrel
yeah.
eye fish.
You know what? Zach knows what I'm talking about.
Yeah.
Yeah, like that one with the transparent head, and you can see like its brain and its eyeballs.
Oh, now I know which one you mean.
Yeah.
Yeah, they look cool.
Yeah, I'd like to see one of those.
And when have you seen the phantom jelly? When was it? Which cruise?
We actually saw it several times. I did not see the first time. I was not on the cruise that we saw it the first time. I think you were, Zach, on Monica Bright's cruise. And then the first time I personally saw one was in Costa Rica last December, so over a year ago. Yeah, super rich midwater life there we found, so yeah, exciting. I think we actually saw it twice in that expedition, so we got to get some really beautiful filming out of it, so yeah, it's definitely a treat.
Matt, here's a new question in the chat. So, what is the process to actually prove you found a new species?
Yeah, that's a long and hard piece of work, because you have to, first of all, roughly identify what it is, what genera it's in. You then have to compare it to every other species in that genera. And often finding the literature, it can be quite difficult. A lot of species descriptions are published in small local journals. You have to get hold of a copy of that. You then frequently have to translate it into your own language so that you can understand what it says. Ideally, you should go to the museums where the original individual is deposited and examine the original type species, they call it, to make sure that the one you have is different. Once you've done all that and a detailed examination with good quality photographs, microscope photographs, electron microscope photographs if you can, and these days often we do genetic sequencing of the potentially new organism. Once you've done all that, you put it together in a paper and send it off for publication. and a group of other scientists will criticize what you've written. Then you respond to their criticisms, and eventually, after a few years, it gets published. And the individual that you use to describe the new species has to be deposited in a public collection, which in Chile would be the Natural History Museum in Santiago. But wherever you are in the world, the species should be deposited in the national collection local to wherever you collected it. So species in Chile get deposited in Chile, species in the UK get deposited in the Natural History Museum in London, the US in the Smithsonian, etc. Yeah. But it's a long process.
It sounds really time consuming.
It is very time
But
consuming.
I actually had no idea that you have to store them in like museums or so. That this is one of the rules.
Yes, increasingly, most countries have a law that says that the original type species should be deposited within the country where it was collected.
Oh wow.
Obviously, in the past, it used to be used typically to Europe and deposited there. But these days, certainly in Chile, the law says type species should be deposited in Chile. So even though colleagues from Europe may take the individuals and identify them, once they're done with that process, the type individual should be sent to Chile for the collection in the museum there.
Interesting.
Yeah.
Where's your RV? Waypoint.
Sí, waypoint.
We have samples.
Matt, do you have an idea why we keep standing or like pushing the central plate up? It looks like they're walking, but they're just like not moving.
Yeah, I've seen them, quite a few of them doing that. I have no idea. I'm not an expert on brittle star behaviour. why that particular species does that,
It's really interesting.
I don't know.
Maybe
It's
I'll
a
have to look it up.
mystery.
Maybe we can solve it.
I doubt it. That's for future generations.
So, my generation.
Yeah. I'll be retiring soon.
So maybe interesting to know for potential future scientists, how do you become a scientist? So I can just talk out of my experience and perspective. I'm still a student. I'm a master's student, but I started studying biology. I did my bachelor's degree, and now I am close to finishing my master's degree. And after that, because I want to become a real scientist, real in terms of I want to publish and go on more cruises, I am probably doing my PhD afterwards. So I will continue doing research, hopefully in the deep sea area, because it's very fascinating to me. And yeah, we'll hopefully get to see more of the unhidden seabeds, and I'll get the chance to explore and potentially describe new species. But yeah, that's kind of the way to go, at least for me. Matt, have you always wanted to become a marine biologist? Was it like a dream job as a child?
For me, it's family business. My dad was a marine biologist as
Oh,
well.
really?
So,
Oh.
so yeah. No, I was always going to be a biologist, but I like plants, but I'm allergic to pollen, so botany was out. And I've pretty much the whole life never lived more than 5 minutes walk from the sea, so it was obvious.
Matt, when did you immigrate to Chile? When did you start working in Chile?
Oh, I arrived in Chile in 1996.
Ah, okay.
Yes, I only came for 6 months, but forgot to go home. So yeah, I've had the opportunity to travel the entire length of Chile and Antarctica sampling. So yeah, one of my research assistants said I know Chile better than most Chileans. So. With regards to where the samples go after we've collected them, well, the ship will be returning to Punta Arenas in Chile. We have permission to sample that will come from INACH, which is the Instituto Nacional de Antártica Chilena. Chile is a signatory to the Antarctic Treaty. They have the power to grant permission to sample. Once in Chile, most of the samples will probably stay in Chile. In Punta Arenas, my samples come back with me to Puerto Montt. Some will go to Valparaiso, others to Santiago. Some of the specimens will be deposited in the museum in Santiago. Some specimens will be exported to, we have an international group of scientists, so some will be going to Germany, the States. But as I said before, if we find any new species, they have to come back to Chile. Although technically Antarctica isn't part of Chile, so so that's a bit of more of a grey area. But yeah, we all share it these days.
So at the moment we can see the seafloor is covered with a bunch of sea cucumbers. So it seems like this area that we are sampling at the moment is like the perfect spot for them regarding the conditions. We're feeding reproduction, potentially reproduction. It's like a sea cucumber garden.
Sample. We're going to get a sack, a bag sample.
Chiquito.
A
Saquito.
Saquito. So, any random piece of mud. Yeah, so we're going to collect a bag sample now, also known as a saquito. Basically, we just collect a bunch of sediment from the surface, and this will be filtered to see what worms are living in the sediment. It's mostly worms and small crustaceans maybe as well.
While we're waiting for the view to clear again, just a quick shout out to my colleagues from the Senckenberg Institute in Germany, Patricia Kaiser and Fritz Schiller. We already can't wait to see you again in a few weeks.
Hi, here Américo Montiel. We are already take qualitative samples with the bag. This bag have the purpose to found specimens to determine the taxonomy of the animals. The procedure of these samples is go very gentle and slowly for the surface of the sediment and take all the animal, because most of the biodiversity are concentrated in the first 5 centimeter of the sediment. Ahora para los hispanoparlantes estamos haciendo ahora un muestreo cualitativo por medio de el denominado saquito, ¿ya? Este consiste en un marco de acero con una lona impermeable, con el propósito de determinar los animales que existen en la superficie del sedimento y para hacer trabajos taxonómicos. El procedimiento de esta muestra es pasar suavemente por la superficie y obtener el sedimento de los primeros 5 centímetros del sedimento, ya que en esa porción del fondo marino es donde se concentra la mayor diversidad. Deutsch? Deutsch? Ja. Now,
Keep moving now, huh?
Although if we see a rock that's gravel,
Sophia. The sea level's not working right now. It's frozen.
Oh, did the VM?
You cannot log the sample.
Let me see.
Bridge, ROV.
Yeah,
Got a new one.
rock is down.
Matt?
New waypoint.
I'll be back. I'm just gonna reboot the. Yeah.
Huh. Drop his contacts. You can totally see through them. Glass fish. Ooh,
It
weird.
looks so cool.
What's that? Yeah, he said he got a new waypoint, right? We lost our track of them somehow. Bridge, ROV.
RV Bridge.
Hey Luke, I don't know if something changed up there or not, but I don't see any more info on the ship as far as the speed over ground or any of that stuff. Did y'all lose something up there?
What are you talking about? The seal is not working anymore.
No, we're trying not to know. But yeah, I don't see that on the green sea.
Yeah. Okay, well just be aware, I don't think I'm gonna be able to see where you are then, right?
Yep, okay, copy that. Hopefully it'll come back soon.
Yeah. Huh? So,
Yeah, the ship is not moving on green sea from the other.
yeah, it's weird, huh? We just seen that. Maybe I'll come just try and restart it up there, but,
For now, I'll look at the USBL monitor and see your position and the ship's position.
okay, thank you, sir.
We're working on
Thanks.
the things to
What do they know about that? The boat's not showing the tracking?
yeah,
Okay.
like a whole bunch of GMs dropped out, so now dropped out, Seawalk dropped out, like a whole bunch. So Coco's on it right now.
Okay.
Okay.
So apparently it's a big thing that's just happened, so.
Yeah. Sorry about that, guys.
Okay. Well, this is not just us being crazy. Other than that, that's been pretty good. No big drama. Enjoy. Okay.
I'm trying to figure out how to put in there.
So that one that's in there, that is USB. I'll get the good relation to the ship on the RV. You read as the RV, is it? On that one?
Yeah, the red, yeah, Bravo is the ROV.
Okay. Alright, alright.
Oh, you're good.
No, you have to look up there. Not as,
Could you switch to USB on that from your thing? Yes. And try to,
USB on this.
yeah. Yeah, try that for a second. Just see what happens.
Ship is currently stopped.
Yes, I believe so. Also, to lock your watch change.
Good, Robbie.
RV Bridge.
Yeah. Just want to, while we get our bearings down here, just want to see if the ship's stopped or not.
I am stopping the ship. Yeah.
Okay.
So,
Yes, flight wall, all item for
so the ship's position is up there.
Yeah, I saw it.
So according to that, the red is SOI while the vessel is the green. Vessel is there. Oh, that's the vessel there. What? Yeah, we just like that. Technical glitch. You want that? Remember that?
Yeah.
ROV bridge.
Yeah, go ahead.
Yeah, Zach coming to the bridge.
Not sure. He just left control M.
Okay, okay.
Could you put that image on the top left on?
Sure thing. Top left, you said? Oh,
Yeah.
top right?
Yeah,
Doesn't
top
matter.
left.
Top left, gotcha. The USBL?
Yeah.
Okay. Let's see, put it here. How's that?
Yeah, that's it.
I don't think so, but let me try.
Get the depth from there.
Yeah,
Go from there.
yeah,
Okay.
yeah.
And obviously the use.
Oh, yeah, nice.
Use
Oh, excellent. How's that?
Better, yeah.
Cool.
Are you guys able to get that through the
No.
No.
It's not up.
Coco's on a whole bunch of instruments and we have them downstairs, so. Unfortunately, it's not something I can fix. So the ship's gonna go for, so this is not.
The RV data is on the ship data. Right? The RV data, oh, the green space is not good, right? The vessel is. Right, it's in clockwise 90 degrees.
The pause is not operational right now, so we're down a couple
Yeah.
nav systems.
So the RVs are these, and the vessels.
I would proceed with caution.
Yeah.
Yeah. Oh, cool.
Alright, just go ahead.
So the ship setting is correct on the USBL screen, right?
Yeah.
As long as I go 45 to the ship, that should be okay. You guys on our You guys on our
Aye, but
We'll get you there.
Yeah, I would, yeah, a couple things have dropped out. I would not trust the ship or the RV position right now until we get those systems back up.
Alright, you.
Sorry about that, guys.
Alright.
I'm gonna land here and I'll just, listen. 'Cause I think- 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 '
So we can go a bit off track. It's just because this from the waypoint up to 10 or 11, sorry. Yeah, up to 11. I think this is kind of a cliff. So we can try to explore a little bit to see that the sponge down there.
Yeah.
So we're in a pretty good waiting for our navigation software. Yeah. We're waiting for our navigation software to come up. So we are.
We're not really moving too much.
Yeah, we're not moving too much right now. Right now we are in Green Sea software. So that search position is not there's turn off the USBL.
But ours is.
Ours seems to be tracking. We put it to USBL too. But only see where our beacon is and ship's beacon. So we're not moving too much until we can get our navigation software. But it just went down. Just went down like. Exact something like your kidney.
Imagine that.
Yeah, yes, I think it was crossed down. But yeah, so you won't be able to see the ships underground, ships leaving.
It's too big.
Yeah. This one is it flatter?
That one I think is better. That one's solid. No, this one's too big. Well, we're not really moving yet until the vessel gets the system back up.
Yeah.
Cooper is working on it, so you could fly around here and look for a rock if you want, but don't go too far ahead.
So we could see how it's going. So the Green Sea software is really gone for the so the data that we can see is beyond. So we're not really the 2 side.
No, probably close.
But yeah, so we're not really moving too much until we get the Green Sea back up. Because the position of the ship this position. Our ROV seems attractive. I had to change the GPS. Because that's a physical piece of stuff. So I think we should go to start right on there.
Can the ship see where we are?
Yeah, the red is us.
So that is us. And then that's about like 75 meters or so. That's us. So we're not really going too far until they get that like a front line. That could or watch that now. Figure out the rock cut.
So he said, don't trust anything.
No, we just want to, it's not necessarily to follow the waypoints again, it's like exploration mode, so.
No, yeah,
Oh,
but
sorry,
right now, there's a problem with vessel tracking.
Yeah.
So,
We could also not.
yeah, it's unsafe for us to be flying around. So we could just look around here to see if we
Yeah.
get a rock, but we're not gonna go to the waypoint again
No.
right now.
No,
But we were
exactly.
we were wondering if we can navigate here and there's
Just,
a wall, you know, like we're facing a wall. We can maybe continue, just have a look around.
And come back to the same.
Yeah.
Just
Yeah, you
have a,
could go.
keep moving, keep looking around.
Yeah, but don't go too far.
Yeah.
Exactly.
Because of the animation, you should see it.
So yeah, because the other side of the wall is something to look at and then we don't.
Yeah. Yeah. Yeah.
There's a little rock. That one should be
That would just be another
There's a ship.
Sounds about
this
that.
can
Then
pour.
probably a bit more better.
And what's on this? Some fucking buddy left there, 35 years ago.
Actually, the vessel, according to that, is more like here.
Sabotage.
It's 96 meters.
Where you are,
15 meters or something.
Scientists. Scientists that risking falls from the
can work.
No, just you earned it. On top and bottom, there's a
This one, Matt?
Small one.
There's a little one just to the above it. These two were too big to pick up.
one, two, little one right there. Leaky. One of them was over.
Put it in our book. Thank you. A little bit extended.
Alright, let me retract a bit.
Inserts up on there.
Someone said that's too big.
These two are both too big.
These
One
one,
and two
two,
egg good ones.
You couldn't get a grip on it.
Yeah, it's too big around. It's like the same problem we had the other day. Slips out.
Capture that huge fish tank. Where's it? Maybe
People that
Gonna come up a bit.
tail. And they suctioned up some different type in the buttons of and catch recorder shrimp.
I don't know. I think it's gravel.
This and sponge. I didn't want to leave the sponge, so it's so very, very, very trying and we just going around the sponge. Pull it essentially.
You don't think? Do you want to try and grab this big one?
If you think you can do it, go for it. But
If not, I don't think so. That little rock is. That little rock's well good. I'm going for this one.
very steep slope, so yeah, we lost the previous one to roll away down the slope.
la tierra.
I'm just gonna push this down.
You want to see it floating, huh? Let's stop. Slide. Pato. Oh, okay, good. Oh, you're all. See.
I was wondering why it was closing so easy.
What's changed?
Thanks. Trying to get a rock. You're going so soon.
Yeah, I'm hungry.
Enjoy your lunch.
Thank you. These too close now. Yeah. We go for a second. Let's not go for a second. Might need to. Happy ending.
Yeah, that's a while.
Zoom in. It's a big rock. Okay. Let's go for something else.
Okay.
There was a small rock. That was part that was molded on.
Hey, are you guys aware that we're still having the network outage?
Roger.
So, I wouldn't trust your nav data.
Roger.
We don't have sea log either, so we're still waiting.
Yeah, we're just gonna move around.
Okay. Cool.
See.
see if there's something. So good morning, everybody. We just had a change of watch. And now in the room is Teresa, Amber, Jessica, and Rachel. We're going to be here for the next 2 hours. Currently, we are at a depth of 520 meters, performing our last dive in front of the George VI ice shelf. Later in the evening, we're gonna ascend and then go to a new area and start mapping, and tomorrow there's gonna be the first dive there. We were supposed to start at a really deep depth, which was not possible because there was a lot of ice over that waypoint. So we started a bit shallower. As I was saying, currently we're at 520 meters and we're coming to a very shallow area, which is at a depth of 270 meters. That's going to be at the end of the dive.
Yes, and some corals and fish. I have to show you your fish. Very cute.
Buen día.
Hola.
Hola, mi amiga.
¿Qué tal, Ben? Bien? We caught another fish last night.
Technique, what?
Yeah, with the nets this time.
You want that fish?
Which one?
Oh, it's gone. That's small fish.
For Tom. I will let
that
you
one,
know.
Ben? You think we can? Yeah. See.
This one?
You think? Yeah.
I think. Yes. I heard you want to talk.
Thank you.
Yeah, we can let the documentarians introduce themselves. They want.
It's about rocks and sediment.
Hi, good morning. My name's Rachel, and I'm a PhD student at University College London in London, England. I'm just coming up to the end of my first year, so it's all still pretty new for me. Quite a steep to vertical learning curve. But this is an excellent opportunity to really take advantage of everything that's going on and learn new things, so it's really cool to be here. I'm a little bit older than your average first year PhD student. I'm 54 and I've had quite a twisty and turny journey to get here. People like were saying whether we're biologists or geologists or things like that, and I'm really not quite sure what I would describe myself as. I did a physical geography degree 30 years ago, so that was where I started. Then I took a lot of time off at home, bringing up a family, and then about 15 years ago, I went back to part-time study and I did a lot of maths, and then I did a degree in astrophysics at UCL. And then from there I went on and did a master's in climate change, and I'm now doing my PhD, which is looking at how the Antarctic Peninsula ice sheet has changed over the last 20,000 years, since the last glacial maximum, since the last time the ice sheets were as far expanded as they get, so when it was really cold during a glacial. So I guess my PhD involves a lot of geomorphology, glacial geomorphology, but also requires a little bit of oceanography maybe, requires a lot of GIS, Geographical Information System, which I'm still struggling to learn, but getting there. So I guess I'd probably describe myself as...
I guess I'm a marine biologist / ecologist. I'm currently a research fellow at the University of Aveiro in Portugal. And here on this cruise I'm working with Teresa, and we're trying to assess the ecological role of holothurians, also known as sea cucumbers, in the trophic chains in the deep sea in Antarctica. So we're trying to collect some sea cucumbers, which we already did for this dive. My work outside of this is completely different. I'm working with fish from shallow and mesophoric zones, and these are fish from Cabo Verde, so warm waters. So I'm definitely learning a lot. This is also my first scientific expedition. So yeah, it's been a great experience, a lot of new knowledge, and I'm very happy to be here. Thank you.
Let's sample the rock.
Okay.
Does it fit?
Out? Yeah. So,
Wow, it's big.
track down the constant. Two
Oops.
That's
Big.
You can see this one on the side. You see it? You see it?
I see it.
I see it.
Just that one there? All the there?
And
there?
here? Is
That
it?
one there? There? You see that?
Seems to be, no?
Place to be there. There we go.
Yeah.
Good job.
We need a picture, yeah. maybe in the baskets, I think.
Outside?
Yeah, I think so.
Okay.
We have some sea cucumbers here. Cannot be.
Outside?
Yeah, I think so. Rachel, as you are the only geologist here, oh, besides Fight is okay. What can you say about this rock? Because for me, it's a rock.
What can I say about that rock? It's hard. It's hard. It's covered in sediment. So it's hard to see much about it. I guess, what can you say? I guess it's come from the rocky outcrop, so presumably it's of the same material. So it's a good way of being able to take a sample back and work out what the dominant bedrock is around here. I think from what they were saying yesterday, I can't say for sure because I can't really see. It's so covered in sediment, it's kind of hard to see. But it looks like that classic sort of black rock that we're seeing outcropping all over the place. It looks like that. And from what the other geologists were saying, they think this is metamorphic rock. So it's originally a sedimentary rock, which is formed by various types of sediment being laid down and then more on top and more on top and more on top until it becomes so compressed that it actually becomes rock. And then to make a metamorphic rock, you take that rock and then something will happen, like some form of folding or faulting, where the ground is deformed and then this rock is subjected to really high pressures and really high temperatures that actually makes it change on a fundamental crystal level. So its crystals actually change and it becomes a different type of rock, it becomes a metamorphic rock. So from what they were saying yesterday, they think it is, the dominant bedrock around here appears to be some form of metamorphic rock, but it would be hard to say exactly what it is until you can get like a little bit of it and have a look under the microscope and see what crystals it has and see what it's made up of. But yeah, generally, I think that's what the type of rock that we're looking at here.
Wow. Quite a nice definition of a rock.
Said
Really
confidently.
Cool. Yeah, in the English way. Very nice. Very nice indeed.
Hopefully correct.
We'll have some room to the left.
Okay. Still not back.
Just magically.
Just put something there and then change it up.
The outer one is 100, you said?
It's about 75. Thin one inside is the 50.
The thin inside. So we
At the moment, we are doing a transect on this wall. We are on a transect mode, so we have our lasers on, and at the end, we are gonna try to estimate the diversity and the densities of the animals that you can see here The same way, we will also try to describe how the sediment and in terms also of sedimentation rates are in this place. As you can see in the image, you have some sea urchins, you have some sea cucumbers from the family Alpadids, some sponges, some fish. Oh, cool. Oh, wow, that's cool. Beautiful colors. It's nice, huh? Gone to the bees.
ROV Bridge.
Okay, go ahead, Rich.
Hey, there doesn't seem to be a quick fix to get possibly out of green seas and sea path in, so we'll have to continue flying by USBL, which means you probably have to give me directions that you want me to go, because I can't see the vessel in relation to your waypoints anymore.
Roger that. We'll plot a plan here. So, I want to add Germany to this dimension.
Yeah, so I would just move the boat 100 at a time.
Yeah.
And then you can look over here on Ranger.
That's what I'm doing. That changed. Well,
Yeah, 500. 500. It's just 500 away, right? So you're in a ring. You're almost 100 feet away from the boat.
That's correct.
100 meters away. Sorry.
That's my estimate. Yeah.
So yeah, you can tell from that. If you guys aren't comfortable, let me know. But I think we're okay to do this.
Yeah. Because it's not too steep anyway. So, Joel, basically, just follow the line, and I will keep moving the ship.
Right.
So, basically, we're gonna head this way, and I'm just gonna keep watching him. And somebody changed the range up there. So, this is the waypoint that we're gonna be using, and we'll just match it with that. Just run a bearing of east for now. So we'll try that for now.
Gonna be ready.
Go ahead and start with this. Bridge, can we make a vessel move 080 for 100 meters? Almost east, east, 100 meters.
100 meters directly east. or did you want to go 080?
080, Phil. Let's do 080.
080, 100 meters coming. Go in, heading 080, 100 meters at 0.4 knots.
Okay. Cool, thank you. Sure. Okay. Ich versuche mal ein bisschen was über die Ausfahrt zu erzählen auf meiner Muttersprache, welches Deutsch ist. Also wir sind hier gerade beim, wenn ich mich richtig entsinne, Dive 786. Das ist der letzte von 4 Dives in der Region, wo vor ein paar Tagen bis Wochen ein großes Stück vom Eisschelf abgebrochen ist, das dann Richtung Norden gedriftet ist. Wir sind quasi also die ersten, die sich angucken, was unter diesem Schelf los ist, was da lebt, wie dort die Topografie ist, was wir sehen an Sediment, an Gestein, solche Parameter. Und es ist eine ziemlich außergewöhnliche Situation, weil eigentlich niemand bisher ein Unterwasser-Ökosystem erforscht hat, was so kurz gerade erst frei ist von Eis an der Wasseroberfläche. Genau, deswegen sind wir total lucky, fällt gerade das deutsche Wort nicht ein, jetzt hier zu sein und uns das anzugucken. Und wie vorhin schon auf Englisch erwähnt, ist das jetzt der letzte von 4 Dives und wir sind gerade in einer relativ flachen Area, gucken uns also an, wie sich hier der Wechsel von Fauna oder von Gestein oder von Sediment von ungefähr 500 Metern bis 200 Metern verändert, wo wir Tiere sehen, wo wir Leben sehen. All solche Sachen sind immer ein bisschen davon abhängig, wo wir Strömungen haben, wo die Tiere sich durch die Strömung eben mit Nährstoffen versorgen können. Das haben wir jetzt versucht, durch diese 4 Dives herauszufinden. Und dann geht es morgen weiter Richtung Westen. Dort werden wir dann nochmal neu mappen und dann nochmal 2 oder 3 Dives dort machen. Und dann geht es, glaube ich, noch an eine letzte Station und dort haben wir dann auch nochmal 2, 3 Dives und dann geht es am 4. Januar zurück Richtung Punta Arenas. Der Transit dauert dann 9 Tage, eine relativ lange Zeit. Hoffen wir, dass die Drake-Passage nett zu uns ist und wir nicht so hohe Wellen haben. Und ja, dann gehen auch schon die 5 Wochen hier um. Es ist immer echt verrückt, wie schnell die Zeit hier vergeht. Ich bin jetzt 3 Monate nicht mehr zu Hause gewesen. Ich freue mich natürlich tierisch auf meine Familie und auch wieder auf mein eigenes Bett. Ja, ich weiß nicht, wie viele deutsche Leute hier gerade anwesend sind, aber ich hoffe, ich konnte ein bisschen Einblick geben in das, wie es hier so gerade ist und was gerade Phase ist. Teresa, now you have to translate everything I just said.
It's limited in German. I only know one sentence.
That is?
I cannot say it here.
Okay. Get it.
It starts with du bist. And then
We don't have
something.
swear words in German.
Yeah, you have it in Dutch, and I know Dutch. Cannot say it.
Swear.
Yeah. Zoom in, please. Oh, cool. Some corals.
Bridge, can you slowly come to an all stop there?
Stop in.
Is it safe for us to sample? Yeah.
Yeah, it's safe for us to
I think because Erin, I think she's interested in one. So if you could, yeah, okay. Okay.
About 60 meters from the sheet. That's not the best one, we'll just keep moving forward. Okay, you want this one?
Yes, please. Erin will be very happy.
And it's gelatinous soft. So, shall we suction it?
Don't think so. Do you think you cannot do it with the hand?
We can, but it might get pushed at the base.
Okay, well, try. If it's impossible, you think, then we do it with the suction.
Possible. I just want to make sure that it's alright with you. So I will cut it at the base.
Yeah.
See?
See. Very confusing, Ben. If you start talking in Spanish or in Portuguese, I might reply in Portuguese and then yeah.
Sophia was requesting for whatever reason to zoom on the horizon at some point.
Yeah, we're trying to park now.
Okay, no, I was just
Yeah.
saying what's in the group. Because that's
Okay.
my job.
Let's see. Where are we going to put this?
In the coral river, they are all, yeah, they are all empty. So you can choose the one that suits you best. Yeah. And this one as well. Different size.
The one on the top, the middle.
Yeah.
That's a picture.
Yeah, that's a good one. See? Good. I glum, yeah. Please. It's really a weird animal. I think. Nice, thank you. They're not feeding anymore. They are stressed. Cool, thank you. The coral. Yeah. Yes. 75. Very sticky. Wow, nice. Mm-hmm.
Nice.
Cool, nice. That was not a challenge. Let's find some fish. There is a competition now. For now, Joel is the best. Let's see. Did you see it, Rachel, last night? The two fishes we caught?
Yeah, I did.
Which one you prefer?
I didn't see the sucking up.
Oh.
I did see the net line. It was some very classy fishing
it was.
action going on. It's like, goodness me.
Tom was very, very happy.
So that was very, what's the word? Dexterous. Yeah.
Amazing.
We're a good team.
Mm-hmm.
Sure. Are we a good team? Yeah. We have a really good team. That's so far I work with Adam as with Joel. Joel might be a good fisherman.
Let's find a shark. We haven't seen any.
So, Sophie, who wants to look at something?
At the browser. This. Just a zoom, please. Thank you.
No, there's a. Is it a polychaete behind? Or is it
I
just a
don't
shadow?
know. Something inside.
There's something behind. But we will probably break it if we suck it up.
Yeah, I think so.
I mean, it's a polychaete. It's on the
It
list
is.
for sampling things.
Okay. Shall we? Yeah.
Are you talking about the pork?
It's a worm here.
The one, if we can sample it. We could probably pick that out.
Yeah?
It's possible.
Okay.
But that thing's gonna swim away though, isn't it?
No.
I mean, if you grab it hard, it can't, but otherwise they're going to swim.
Yeah.
Try. Grab it.
Mm-hmm.
Grab it, suction.
Grab, but hard.
Grab it. Grab the.
Oh. Yeah, maybe with the suction then.
You just want the animal, not.
Yeah. It's
Yeah,
a
it's very suction.
yeah.
Which they're all empty. So
Yep, yeah.
the big one would be 5.
Yeah? 1, 2, 3, 4, sí. 5? Show me.
Yeah, because yes, 5 is bigger.
That's my size. Okay, let's go down.
We're going to this the last one. So when you spin the suction, it spins fast. And then once it gets up in, slow it down again, right? Slow it down to about 1.
It goes to number 5, yeah?
And sit as well.
5. Yeah, 5. Jéssica, como é que pode estar com sono? Não estou a perceber.
Do some sits. Maybe more. Okay, that's good.
Right, ready for.
He's already ready.
Nice. Médico. I say the gin tonics. It's cool, actually. Cool. Yeah. Good.
Thank you.
Thank you, Ben.
It's Joel. Okay, so should I index to number?
Yes, please.
Index to 6.
Is there anything else?
No, it's okay. Thank you.
Suction stop.
Perfecto.
I asked a gin tonic to America for this sample.
Gin tonic?
Yeah, they're recent. I like it a lot.
Suction in.
Mm-hmm. G&T, Rachel.
That's one thing I don't drink, which is not very British.
Oh, yeah, you don't, yeah.
No, but even when I did drink, I didn't drink G&T.
Oh,
It's more Jack Daniels and Coke.
I'm very British.
Anything else from here?
Nothing. It's yes. Thank you. You don't have yet. Still flying blind.
Keep moving direction. I'll catch up.
Okay. Let's see.
Bridge, ROV.
Go ahead, Ben.
Yeah, Phil, we're gonna move ahead and just stay where you are. We're gonna get to about 100 meters away from the vessel, then we'll go ahead and proceed with the move.
Yeah, just give me a shout when you want me to move.
Roger that, thank you.
Muito boa tarde a todos. Desculpem o meu silêncio, mas estávamos aqui num momento estressante. Estivemos a mostrar primeiro uma rocha, depois um coral e agora uma poliqueta, ou melhor, uma minhoquinha do mar. Neste momento estamos de volta a fazer um transecto. Como vocês podem ver, temos os dois lasers que nos vão indicar ou que nos vão dar informação sobre o tamanho do animal ou dos animais que existem no fundo do mar. Neste momento estamos mais ou menos a 480 metros de profundidade e pronto, estamos a tentar aproximar de um platô que existe em cima desta pequenina montanha. Podemos ver holotúrias, basicamente dois tipos, uma muito provavelmente é Scotoplanis, ou melhor o porco do mar, e a outra é que vocês estão a ver neste momento é Peniagoni, ambas da família Peniagoni, Elpadites. Vêm também ouriços, briozoários. Where? Huh?
Anyone there?
Where? Here.
Let's do it.
It's too small. Competition.
No, there's an octopus on the right, but a tiny one. There's an octopus on the right, but it's tiny.
Oh, yeah.
But at least it has its arms out. It doesn't look completely sleepy.
Wow.
There's some interaction.
The Allosaurian and the octopus. No. Then you'd kill me. I won't, but
No, it's just nice to look at.
ah, very cute. That's not Dumbo, this one.
It looks like
Wow.
it's like
So
glowing. and-
cool, yeah.
Wow.
Oh, now it's changing.
Really?
Oh, it's so cool.
Ah, damn.
You never had problems with octopus, Ben? While diving? Yeah. Sometimes they come to you. They grab you. Mm-hmm. It's one of my favorite dishes.
Jessica cries. I will tell Tony to make a good picture.
I didn't see it in Mexico. Yeah? You have?
Especially in Yucatan.
Ah.
Yucatan and Campeche. It's very rocky.
Yeah. They are big ones or they
Yeah,
tiny?
they're all those sizes, but they catch
Cool.
them a lot. From less for Yucatan.
As I was only in Chiapas. I fall in love with that place. It's amazing. Not touristic, not too much. Yeah, it was cool.
I've driven motorbike there. Chiapas to the lakes and mountains.
Yeah, I went from Chiapas to Oaxaca by car, by bus. My God, it's beautiful. Yeah, sponge. Yeah, it's coming right.
Bridge, can we make a move? 50 meters, heading east, 090.
50 meters, 090, coming.
Grazie.
Parli in italiano, Ben? No. Sì. Beautiful. Bello. Yeah. Yes. Yeah. Thank you. Different kind from the other ones. Some rocks. Can you zoom in, Joe, here? I think it's the same species. Yeah. That's the same thing. It's okay.
Those nerves.
Nice. Good, thanks. Nothing happens.
Someone in the chat asked, what is this?
A sponge.
He was referring to the sponge.
Yeah.
I was wondering, what do you mean?
It's only sea cucumbers, basically, and sea urchins.
Something. Is that a sponge on the left?
Here, right?
Oh, okay, yeah.
Just a zoom. Yeah. Cool. Interesting. Well, no, it's okay.
No, I think it's an anemone.
Yeah, it's an anemone, I think, yeah. But it's closed. Yeah. I'll zoom in this for Anna. A cup coral. Thanks. Yeah, that's good. If you want, you can answer. I think it refers to the ice sheet. I think.
So yeah, I feel like answering this could be a bit of dangerous half knowledge, but from my opinion, it was closed off for a long time. Like people were saying at least since the last ice age, I guess longer than that, because Antarctica has been quite covered by ice. I cannot really give you an exact time scale, but at least minimum 20 to 30,000 years, I would say, if not longer. But what you definitely see, I mean, we were not really sure what to expect down here because we have a week or 1.5 weeks of exposed sea.
Oh. Ben, a challenge. A fish.
A fish.
No, really.
Is the net still there? Don't
Yeah.
go near it. Don't go near it.
Because Tom, yeah, He doesn't have this one.
The net is on the outside.
So yeah, but what we're seeing, I think in the first dive there was less life, which we thought would be due to the ice sheet. But I think the more dives we do in this area, the more we see that there's actually a lot going on down here. So some explanations could be lateral currents, I guess. So not having this kind of marine snow from the surface, but definitely having currents that are coming from the sides, transporting nutrients in the area that is under the ice sheet. Because we definitely see life here, and we see ecosystems that are functioning, and we see a variety of different taxa. So there must be some transport of nutrients to this area without having the necessity of having the deposition of nutrients from the surface. So yeah, I don't know much about cave or island type ecosystem, but I guess you're referring to something that is kind of shut off from outside environments and develops their own ecosystem for various reasons. I mean, I would say that what you find here is definitely also the taxa that you find in areas that are that they're not having ice sheet above themselves. But I could also imagine that you definitely have other taxa developing better or being more abundant in this area than others, which can probably be due to more resilience in times of nutrient lack or some sort of that.
Let's go into the rocks. It's clever.
Yeah. It's going away. I'm following the fish.
Yeah.
It's
I'm
going
also
away.
still there.
Still out there.
Yeah. Oh. I don't see it here. Oh, I lost it.
Still there. Still out there.
Oh yeah, there.
See him?
Yeah. Yeah, here.
Let me get the same position.
Where's the plant coming from? Above or behind?
Okay. Stay where you are now. Just,
It's going away.
It's a beautiful one.
Still there, still there, still there.
It's there, yeah.
It's just on the.
Yeah, it was there, here.
Using the camera. That allows Sophie to run the camera.
Yeah, I'm not.
I don't see him.
Sorry, someone had dropped a mud bag,
I don't see that. Do you see it?
No, I'm trying. Yeah.
Yeah.
Wow.
They're fine and all.
Yeah.
There's too much mud in the mud.
No, it's an Holothurian.
Maybe we grab this for a moment. Can you sit there for a moment?
Sitting down. Okay. Happy?
Let's not put this in the back. Change course. Let's put this on. We'll put this past. We'll put this back in the basket.
Is there a fish there?
Yeah, but it's the same kind. EO caught yesterday.
No,
Yeah.
that's a
This,
tripod.
yeah, we caught them, or Joel caught it. This one.
It's a lovely fish.
Yeah. Ah, it doesn't have blood. It's amazing. Yeah. It's very muddy, huh?
No, there's mud in the end.
Yeah.
There's another fish there.
Hmm?
Benthic, see it? The fish.
Yeah, but I can't do anything with that. Just keep your eye on it.
Where?
See if I can
Sorry?
get some on the other side.
Ah, yes. Yeah. Good, good.
Okay, it might be. This might be good. Where's the fish?
Yep.
See there?
Yeah, it is.
That one? Okay.
Mhm.
Let me give it a shot. Is that the one you want?
Mm-hmm. Yes.
Okay. Give it a shot.
Let's go.
Okay, go for it.
Sophie, can you operate the camera?
Yeah.
Just follow the fish. That's the target. I'm going to bring my net up so you can get down low.
No.
That was a good move, but didn't get it.
It's a fast one, yeah.
That's still there.
Yeah, you saw?
I need to regrab this thing.
You want me to bring the porch out so we can put it on the porch instead of the boat?
No, just put it on the ground. I've got all the rocks out there.
What's it?
What is that?
I think it's Ambalula. It was Ambalula. A coral.
Let me know when you're stable.
Stable. Not ready. No, never mind. Stable.
Flying around with the fish.
Good? Yeah. I'm changing my clothes now.
Roger.
Gonna run it all.
Wow, cool. Yeah. Nice. It's beautiful, wasn't it?
Mm-hmm. It's big.
Yeah.
You know what I mean? What you're saying? Yeah.
Cool.
We're just exploring this.
Okay. Yeah. You want to talk then? Sit down.
So we're gonna stop for a few seconds, for a minute here.
Mhm.
Take a break.
Okay. Wow, nice.
Tony's going on a boat again. Tony's going on a Zodiac again, and they're saying
Why?
they have one free space.
But why?
I don't know.
Oh.
Whale in our shift. Boat in our shift.
Tell them that we want to go. Or I want to go. Yeah, cannot talk for everyone.
Possible.
No. Okay.
Wow. You know the species?
Hmm?
Do you know the species?
Of the hydroid?
Uh-huh.
Oh.
Nice.
See? It's a hydrozone.
Go for it. Yeah. Oh, the shrimp.
Where is she? Oh, I see that.
Wow. Wow, so many. Are they gone? Are they gone? Don't know.
Who's gone? Sorry.
Tony and the other.
Yeah, I guess so. I mean, I guess they're about to launch or something. I guess it's for Brody. I don't know what he wants.
Ah,
Scan
nice.
another iceberg.
One more, Teresa.
Mm-hmm.
I'm gonna put this on the side.
Ready? Come on. Okay, so you want to take off, huh?
Yeah. Bridge. We're gonna make another move. 070. 070 for 50 meters. Bridge, are we?
Yeah, go. Abandoned ship.
Bridge, are we? Bridge, are we? So, favor, can you call the one flag?
Sure.
you getting us on clear phone? Ah, okay. Ben, call him again.
Really, Charlie.
Hey, Ben.
There you are. Anyway, can we make a move for 60 meters, 60 meters, bearing 070, 070.
60 meters, 070.
Roger that, thank you.
On the way at point 4.
Grazie, grazie. Yes.
Hey, Joel, do you mind to put the lasers on?
Oh, yeah.
I'm not sure if...
Lasers on.
Okay, thanks. Hmm? Yeah. Ben, do you want to grab this rock? It's a rock.
Sure. Good shot. Put it in the net.
The net. Together with the fish. I don't think there are more rocks.
No.
It's just video transects, right?
Right.
Oh, the sampling is only 13. Okay.
But I guess there's already discrepancy between the waypoints there and the ones here. So right
okay.
now on this map we are at 10, but I think if you look here, it has the opposite shape. So here we are in 11.
Okay.
Going to 12.
So remember, mental mode, not put anything behind the box.
What is that? Sea spider or just a...
Where?
Maybe it's just a ophiurite. I don't know.
Can
But it
you
looks
zoom
very
in?
spidery.
Yeah, it could be. I see spider. Yeah, maybe.
Yeah, it's one.
Yeah.
Cool. Can we collect that?
You want that?
Yeah.
Oh, okay.
We want that, no?
See?
Okay.
You want me to mark that?
What I can say is
you? Ah. No sabía.
We'll just do a slight suction.
Yeah.
That's for everything. Very slow suction.
No?
Sí.
Ah, okay.
Drop this.
Estamos a tentar também caçar peixes para o tomo, porque ele não tem muitos, percebes? Isto pode ir para o 5 ou para o 4? Ou se calhar que este não. Pode? Nós já temos lá estas. Ah, nós temos uma cena aqui. Sim, sim. Sim, 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 sim.
So now it's in 6.
Nós temos uma paliqueta no 6. Pode ir para lá? Pode? We can put them in 6 then.
Okay.
No. I mean, Tony was asking. No. Just take a camera. Number 6?
Yeah.
Still not that good job.
I'm gonna plug this properly so the suction will be stronger. See the suction.
Yeah. It's a different one, but yes, let's go. Have you seen it going on?
Usually I pick up the spiders. For some reason, I don't know why. Can you slowly move forward while it's going?
For the fish? What about the index, but the spider still on it? Hold on. Okay, I'm moving.
It's going away. Slowly. there. Yeah.
Teresa, did you see the question in the chat?
Hmm?
If we release them back into the ocean.
Yeah, I know.
Do we?
No.
Ready? See it? Go for it. Oh, goes to the boat.
Ah. It's hard.
We probably need to
Still there. See it?
No, vai. Sì, Jessica. Aproveita.
Closer.
Ui,
Oh, he's blind.
quase.
He's blind. Follow the camera. So he can follow the camera.
Where did it go?
Mm.
Might have to ram out a bit.
Suction still on, by the way.
Yeah. Okay. Now, see if you can ram it.
On it.
Turn off the suction. Let him relax for a moment. And then slowly turn it on.
So that is the vessel moving?
I stopped. I'm still on 109. 110 meters.
Gotta move it.
Okay. Try it again. Suction. Full suction.
So replying to the chat, yeah, unfortunately we do not release the animals nor the rocks back to the ocean. Even if we would do it, yeah, they would not be in a very good state because these animals, they are collected. They have been collecting from 800 meters, 400 meters, and then back to the surface. So I would not think that they. Most of them did not arrive, well, alive. But we are not collecting everything we see. We just collect what we actually need and the minimum amount so that we understand how this ecosystem is working. So in a way, it's for a good cause.
Can we go down a bit, or is it for a specific reason that we're so high?
Yeah.
Thank you.
Bridge. Can we do another 50-meter move? 070.
50 meters, 070.
Should be around. Phil, can we make it 70 meter move, 70 meter move.
70 meters, 070.
Can you zoom in as well to this area? I think it's only greenoids. No. And coral. Okay. Ah, there is a fish pen here. Maybe it's too hard, I think, no? Yeah.
Ready? Go.
Ay, cuál.
It's gonna have to land it on
Right.
Very quick. Wait.
I need to regrab this one.
Put it to a start.
I'm just gonna put this back in the basket. This.
Okay. It's okay.
Fix this. Yeah.
It's good.
Fix it.
It's full of mud. Yeah.
Can we back
ROV bridge, we'll have to stop here and move to port side for a short moment because there's a growler near the umbilical.
Okay.
Area Bridge, would you be happy for us to move 20 or 30 meters ahead to get clear of that growler, at least from the umbilical?
Yes.
Yeah, copy that, no problem.
It's actually change. He said he wants to go forward now. Wants to move ahead.
Another one. Cap coral, yeah.
No.
Ben is gonna teach me though. They are asking if the science are able to pilot SuBastian. If needed, they are asking if we are able to pilot SuBastian.
Scientists?
Yeah.
You won't be the first.
Ben has doubts. So they switch out every 2 hours. I think then they take a break and get some food, and then they get back in after 2 hours, rest. We also do 2 hours, the scientists, but then we have 10 hours in between. So we have way more time to rest. And,
No,
get some
I
sleep.
don't.
Normally, that should be the case. I know you're in a different position.
Yeah, and I don't think that any of us would be able to pilot SuBastian if needed. I'm like trying to think of a situation where that would be needed to maintain the ROV. I think in that case the ROV would probably ascend and not be operated anymore if for some weird reason all of the ROV people are not available. So yeah, I don't think that's gonna happen.
You'd give it a try. It's a joke. I'm so sensitive.
Hey Phil, can we do a 50-meter move, get ahead on your present bearing?
Yeah, that's perfect. 50 meters dead ahead.
Is it possible to zoom in? Yeah. Okay. It's bright as well. Okay. Yep. Yeah, thank you.
Fishing net. Small fishing net.
Hmm?
It's a small fishing net.
How far is it? What's it from us?
97 meters.
It's pretty very small. Full bar, but I don't know if I should not far away from this.
We're just doing small moves right now. It's cattle track. I'm doing a guessing game.
We're gonna have to be patient.
Should still be good. It's 95 meters.
Because every time I go, some of us have this.
Okay. And you need to calculate, right, with the ship?
Yeah, because that's not a good decision.
No.
It's
there.
hard to make this one.
After each.
What's going on over there?
You read that one. That each.
What?
What's that one on this case?
Why are they always facing in the same direction, or is it just random?
Oh, it's maybe the food? Probably. I think the organic matter is, look at the current.
Mm.
It's like that. So that's why I think they are facing.
Okay.
But I don't know.
Looks like a stampede.
Huh?
Looks like a stampede of sea
Mm.
cucumbers.
Yeah, it means that the food here is very good. Sorry, that?
They're not different from us.
Yeah. No, they don't care. And their guts, they are full. Means that they are eating constantly. Cool. And here it's the gonads. They are developed, so it means that they are in a reproductive phase.
And what is the lesson in that?
Ambulula, just to close, a close up. I don't think Erin wants. Yeah.
I mean, there's some stuff sitting on top. I don't know. You want that?
Where?
There's some small amphipods, but I don't know if
You want? No? Okay.
I'm gonna put down.
Yeah, you can.
I'm happy.
Yes, thank you.
Bridge. I think we'll do another 50-meter move. Same heading.
50 meters straight ahead.
Roger that.
Boom, underway.
The fish is on top. Quase, quase. Nothing changes.
Yeah, but I mean, there's way more stuff to see even here in the sediment than there was before. So.
Huh? A rock.
Just a thought, but do you think over this wall we should maybe close a skin or something? I mean, we did one so far, so I don't know. I know we need to reserve, I think, 3 of them. 1 for the push course and 2 for the end for the ascent.
I need an iskin, but when I collect corals for me and
Can you back up for a moment? I want to get a bigger picture if it swells very high or not.
I just want to take over in 5 minutes. Yeah, yeah. I didn't take any because it was only sediment. It was not very interesting for her.
Bridge, can we do
It's possible to zoom or
All stop.
see what's there? Because I think it's a coral. They are, but I'm not sure if they are alive. Hmm? Hmm. Yeah, they are overgrowth. Mm-hmm. Yeah, not good. No. No, they're dead. Thank you.
Too late.
Huh?
You got here too late.
Yeah.
Yeah. Ready?
Ready.
I was just about to say neither Anna nor Aaron are here suspicious.
Okay.
Maybe they're sleeping.
Maybe we close then a Niskin bottle.
Yeah, I would say so.
Hello.
Hello.
Can we close one of the Niskin bottles? It's a different environment, so. Mm-hmm.
Right up on the rocks, just cheers flag.
Yeah, on the rocks. Eden, hello. I'm just asking to close one of the Niskin bottles because it was only sediment during our dive, and now it starts a rock.
Okay.
So yeah, I think it's a change in environment.
Cool.
I collected one coral for you.
Okay, thank
The
you.
white one.
The white one?
Yeah.
Alright.
But other than that,
Thank you very much. Teresa, stay out.
yes, I know. That's great.
Oh, the skin one fired.
Thank you.
Niskin, which number was it? Sorry.
Uno.
Uno. Okay, perfect. UTC.
So I hope you get some things.
Yeah, UTC time.
It's not.
1702.
Flocculent, isn't it?
Yeah, it's like shells. It's like little shrimp shells.
It's little exoskeletons.
Is there some monster up there?
388 meters.
Appreciate it, buddy.
We made it to Waypoint 11. Hi everyone, we just did a science party change, or we're in process of a science party change. My name is Erin Easton. I'm an assistant professor at the University of Texas, Rio Grande Valley. And this cruise is the Climate Connections at the Ice Sea Interface cruise. And we're currently exploring a newly exposed area of the seabed after a chunk of ice shelf just broke off a week or so ago. We're going to start the dive at a little deeper than 1,000 meters, but I believe we ended up starting around 750 meters, and we're planning to work up to a shallow point around 270 meters depth. I'm here also with the partial support of Ocean Census, and this cruise is part of the Challenger 150 initiative. Let's see, we have Tyler and Zach flying right now. Hey! Tyler's flying, and Zach is at the minutes. Soph is on the other side streaming. Moritz is coming in for logging. And we still have Amber here. Although, if you want, I can watch the chat for wish list. Anna can't be here.
Oh, she can't be here.
Yeah, she can't be. She'll come in like an hour, but I can.
Whatever you prefer.
Yeah,
Like,
it doesn't.
whatever you're happy with.
Okay. I don't want to keep you here extra, so you're welcome to hang around if you want, but you're welcome to head out. I'm comfortable with either. Alright, and then we have Rachel as well coming in for logging. So we're just moving up slope.
We look like we hopped on greensies again.
So it looks like what?
Looks like we have you on greensies again here.
Oh, magic.
Looks like we're doing it pretty well too. It's about where we would expect, so good news on that.
Alright, let's hope it lasts.
Okay, so we're moving up this rock face
You get back in here.
here. It's been quite sedimented up until now, from what I understand. We're seeing here some urchins and sea cucumbers. Looks like they already did their sea cucumber collections for the day at the deeper part of the dive.
Guess I'm yes, it's still ground fault in there. And yeah, yes, it's probably still ground fault.
It's still covered. You can see it. You can see it on that of the meter. But it's a little funky. You got a chance to look at it.
So from 11:23 to 2:04. That's how long our switch was on.
How long?
From 11:23 to 2:04. 6.5
I
hours.
wonder how they did to fix it.
Cocoa.
With Erin?
So does that mean we have like sea log and stuff back?
Yep, I just
Alright.
checked.
Awesome. So did you hear? We can sea log again.
Yeah.
Yep.
Yeah.
It just opened back
Amazing.
up. Yeah. Perfect timing.
The log as you want.
Yeah.
Yeah. Yep. Yeah, we had a little tech outage today, and it appears to have just been resolved.
And our overlay's back, which is good.
Yeah. Wonderful. So it seems like quite the abundance of. Ooh, can we go look at that? I'm sorry. And that's like off to the left, Tyler. Yeah, thanks. Maybe that might be a sponge. might also be a coral.
Is that primnoid?
Yeah, I'm thinking it might be a primnoid, yeah, which we haven't seen in this area. We were seeing it at other stations, so if this is in fact a coral, we'll definitely collect it. Oh yeah, it is.
It's cool.
Yeah, so we can sit down, take a good image of it, and do a collection.
It's gonna go in
Coral quiver.
Quiver?
Yep, quiver of your choice.
or my choice.
Yeah.
Best one.
All right, sounds good. Yeah, Andy was feeling all like challenged the other day and started from the back ones. He was like, I'm gonna start with number 7.
It's part of tradition. Tradition is you gotta mess up the next guy.
I know, he was feeling very generous. So yeah, we're just gonna sit down and collect this primnoid coral. So I don't know if they've been seeing them on the dive today, but we haven't seen any primnoids that I'm aware of under this recently exposed area from the sea ice. We were seeing them further out at our other stations, but we all of a sudden stopped seeing primnoids and bamboo corals. So I don't know if there's a depth difference, some current pattern, but it's interesting that we're finally seeing one again. Let's give it a nice zoom first. Okay. And we got a little crustacean on it too. Lands right in there.
Oh, we got cool isopod in there
Yeah.
too. Ghosted looking at that.
He's like, I'm pretending to be a coral. Another one up there. 3 of them. All right. Thank you. And we can take kind of that upper half of it. Maybe trying
The
to.
top's like down, right?
Yeah, the top is down. Yep. Nice, thank you. Just give that coral a little trim so it can keep on.
Want one more glamour shot here?
Yes, please, another glamour shot, thanks.
Oh, I got your isopod.
Mm-hmm. Nice. All right. Yeah, that's good. You can go ahead and put it in. The polyps are pretty small on this group, so it's often hard to get a nice view of the polyps and the skeletal elements on it.
Amazing. Thank you. Little shrimp going by. A couple of them. So yeah, on this expedition we have a number of different scientific teams, from people doing geology and biogeochemistry to oceanography, then those working with the fauna, from the fauna that live in the sediment, two of the smaller size classes, the meiofauna and the macrofauna. So they're looking at worms, copepods, all the way up to polychaetes. And then we have those working on the megafauna. So we have someone who's working with sea cucumbers, I'm working with corals, and we have someone working with environmental DNA and microplastics. So it's quite a diverse group of scientists on board. So I guess back in transect mode as we continue along our waypoint. Waypoint 11. So they want us.
Cone shell.
Oh yeah, can we give that a quick zoom? See if that's what it is, or if it's a dead. It looks like it's okay. We're good. It's long dead, whatever it is. So, keep our eyes peeled for a living one. A nice sea pen coming up. A lovely field of sea cucumbers. All right, so we have our time still to get to 12, and then. Okay.
You guys looking for transparent fish?
Translucent
fish? Is that what this guy is?
It is a translucent fish. I know we're looking for snailfish. The sea pens that we're seeing here look different than the ones.
La primera anistia fue la que tiraron recién.
Sí, fue a UTC 1702. Yeah, let's go. Okay, there's an Anthomastus coral. I think they've been seeing those. I'm thinking that's what they collected earlier today. Let's see. Still seeing some scallops. Some nice sponges. Are those like a line of shrimp on the shelf?
Check out the shrimp on the shelf.
Shrimps on the shelf. There's some brachiopods as well on this rock. 2 shrimps on a shelf.
That's topsides perfect for back.
Yep. Awesome. What you have to do?
Look where to reset the switch.
Oh, a few more than 2. And a little urchin, a little mini urchin, all decorated with a shell piece. Nice.
Cozy little nook.
It is. We can take the lasers off for a moment. You wanna? Get a nice zoom of our nook full of shrimp and urchins. A brittle star there, brachiopods. Miniature. I love the little baby urchin with its own little decorated shell. lasers on and continue transecting.
Is it on?
And so many sea cucumbers.
We'll move yet or wait a minute? Sorry, time for that. We'll move yet or wait a minute?
Probably get them going.
So Maritza, do you think the sea sea pen that we've been seeing is the same as what we were seeing at the other spots?
I don't know if it's the same.
Yeah, it's like seems more dense, like more tentacles than the other one.
Maybe
Yeah,
we
wait, Paul.
can zoom on the polyps.
Okay. Yeah, next sea pen we see, we'll do a zoom and see if we can tell if they're different than the ones we've been collecting on the last few dives. This one just seems to have like a thicker base and more dense polyps. Yeah, it's amazing how many sea cucumbers there are. Little fish sleeping by that one. Crinoid down in the bottom left corner. Wow. So how's our ice situation been today? Is it staying largely clear, or have we been trying to maneuver around the ice?
I haven't had too much trouble about it. I know we started it and not at the place we were going to start it.
Yeah, 'cause there was a huge iceberg sitting right over where we wanted to start the dive. Yeah.
I skipped to the first part, right? Now it seems like it's pretty fun.
Okay, good to know.
Look, you see on the radar, it's a lot of them. Yeesh. It's like a video game, you know? Asteroids,
Yeah.
the final level.
Yeah, there was that one stretch when I went up to the bridge and looked at the radar. So many icebergs around, just everywhere.
Yeah, it's pretty kind of crazy to be diving in an ROV and between all that.
Yeah.
Kind of cool too, but great.
Yeah, it makes for a pretty amazing view. Sometimes they pass so close to us, just like one after the other, as we're maneuvering through the ice.
Those seals look so comfortable.
Oh yeah, they do. Do you see the one the other day where there was probably like 4 dozen seals, like around 50 seals laying on this one iceberg.
It's
Kind of like a
already on like hard, cold ice, but they're
Yeah.
like the most comfortable in the world. Like,
Mhm.
it's a memory foam mattress. And
Yeah. They kind of like roll over like a cat or a dog, like, yeah, this is great. Oh, that's a pretty fish. It's like, here I am. Yep. Yeah, if we can go ahead and give that fish a nice zoom. Oh, there's 2 of them. I missed that one. But yeah, the one that just came into shot though, I think, yeah. Oh yeah, turning. Get the lasers on it, and then, yeah, you can turn the lasers off for the close-up.
Oh, you got like a scar or something.
Just a little tooth inspection.
What? No, I'm getting seasick. Thanks though.
Worth a try. All right, we can continue transecting. He doesn't seem to be want to be very cooperative. He's like, yeah, I want a picture, but not for that long. But there in the sitcam on the right, looks like there is a sea pen coming up. So if we could zoom in on that, go ahead and put the lasers on again, get a good size on it. Thank you. What is the map in the upper?
Oh, I think I just had the radar up.
Oh, you have the radar up.
With the
Okay.
ice.
Oh, okay.
Icebergs.
Yeah. So yeah, I think either I missed the comment or the comment's coming in a little bit late. We're a little ahead of you. But radar was up. Yeah. Yeah, it's different, isn't it?
Más pequeñitos, el otro que era como más.
Mm-hmm. The surface, so you can see where the icebergs are. That's what the radar was of. Yeah, you can go to sit down. I think we're going to collect this because it's different than the other ones we've collected. We're just going to sit down and collect this sample, do some zooms before.
Okay.
Alright, I think you just got the lasers on it pretty good, so you can go ahead and turn them off for glamour shots. Look like there's little isopods or something on the tips. After you're done with the tentacles, can we look at the base where they attach, where the polyps are going in? get a close up there, so just pan up a little. Yeah.
There?
Yeah, right there. Nice. That's some beautiful zooming and cinematography. Oh, yeah. I don't know if it's like a newly budding polyp there, a couple of them, or if those are ones that were broken off. Thinking maybe newly budding ones. Amazing. All right. When you're happy, Soph?
Happy.
All right. So we can. Yeah. So. Yeah. Let's see. How big is it with the? Only one laser showing right now.
Oh, pretty big.
There we are. Yeah. So a BioBox 1 ARV.
So pretty much the whole thing's what we're taking.
Yeah, we'll take it from near the base. Do you want more before we?
If I grab it at the base, you think I can just pull that out of there?
We can, but Alex might want some.
Just one wide shot. Wonderful
Yeah.
to go with that amazing footage. If it's a top possible.
Yeah.
Wide shot with nothing.
Let Alex direct you. We can take the lasers off and see what he get a wide shot of it. Yeah, you know, I don't. Maritza, do you have any idea how old the sea pen could be?
Creo que no hay estudios como sobre la edad de las plumas porque tienen que hacer estos cortes en el esta parte de aquí, en el raquis. Entonces no hay mucho estudio, al menos de la umbelula, solo de la virgularia, si no me equivoco, en el océano Índico.
Okay. Yeah, so Maritza's saying that there really haven't been any studies. So they have to, in order to age it, they have to cut all the peduncle. Yeah, so you'd have to get like the whole like stalk of it and then cut that into pieces and use that for aging. So yeah, they don't really have, there hasn't been a lot of studies, so there's not a lot of information for us to give you a good estimate. There's like one species of sea pen that there have been some studies with, but it's not this group. So this is an umbellula species. Oh, for once, like the sediment's flowing in like helpful direction.
Yeah. So it's a little bit less weary to move. Something get blown away from us, which is nice.
Yeah.
Also, maybe the cat's off. One stronger than the other two. That can be a little bit of depth.
So we're just getting some glamour shots here, working on the lighting.
One final thing. I think I'll put in a box for now. Need to lock the focus. Focus with the mind. Yeah. Focus. And then what happens? You got to leave it as it was actually. Yeah.
some fancy lighting on the coral here, so we're going to be bringing the lights back on.
Wow!
Take some copyright inside our sounds.
Okay, awesome. All right, yeah. So we can, yeah, Zach, we can take it from the base then, and then bring it up from one more glamour shot before putting in BioBox 1, A or B, whichever is easier.
So I don't need to cut it, I can just pluck it basically.
You should be able to like pluck and pull. I'm not sure how deep it's gonna go, but
Yeah, give me a little closer.
so we've collected a few sea pens on this expedition, and a lot of the collections we're doing are going to end up in a museum, hopefully part of a new exhibit in Chile. So we're collecting some representative fauna, and then we also take just a little bit to do some genetic analysis, but with them being at the museum, we'll be able to do additional analyses later in terms of looking at their morphology or physical characteristics, so that if any were to be a potential new species, that the work could be done to describe potential new species, and comparisons can be done to and comparisons can be done to other species and other specimens. So.
Alright, where's my note?
El pedúnculo de esta también es diferente.
Yeah, the Funko is different, yeah. It's got like that thicker base
Sí, el
before.
otro como si era como aglobado.
Yeah.
Como que tiene un anillo arriba.
Yeah, it definitely this specimen definitely has a number of different characteristics that we're noting that are different from the other collections of the sea pens that we've done on this expedition so far. It has more dense polyps. The base, what you can see right at the summit interface, is larger and almost this like larger ring shape. So, a number of different characters. I think Marit's also noticed something different about its tentacles. And I think that's good if you keep moving where that, yeah, it's that back corner. Yeah, I mean, it's the problem with the sea pens. You end up having to often end up kind of closing the door on their stalk. Yeah, go ahead and move. At least it's out of the way. UTC 1736, 365 meters, sea pen, Malula. Alright. No, I don't believe that sea pens gather any nutrients from the sediment. They're filter feeding, taking the nutrients from the water column. They're mostly using the sediment and their peduncle, the base of it, as an attachment point so that they can stay in there against any currents that come through. Thank you. We can go back to transecting mode. So Yeah, of particular interest on this expedition are polychaetes, mollusks, corals, sea cucumbers, and isopods, amphipods. Are some of the fauna that we're particularly interested in. So Maritza actually did her undergraduate thesis research on sea pens. So Maritza, do you want to tell us a little bit about sea pens? Spanish or English, your choice. Is that another sea pen? I'm sorry, can you?
Bueno, los cruceros que hemos realizado, si no me equivoco, son 6 familias las que hemos encontrado de plumas de mar para Chile. Son los primeros reportes casi de algunas, no de todas pudimos tener muestras, algunas solamente fueron imágenes, lo que igual nos sirve para al menos tener el registro de que existe la especie acá en Chile. Hubiera sido genial tener las muestras, pero no se pudo por diferentes dificultades para colectarlas y también el tiempo que uno tiene determinado acá en los dive. Así que tenemos del género Penátula, Protoptilum, Balticina, Funiculina, Umbellula, que es de este mismo que estamos viendo en la pantalla ahora. Pero al parecer las que colectamos en los montes submarinos son diferentes a las que hay acá. Así que ahora estamos trabajando para poder identificarlas y tener ya al nivel de especie identificada de estos organismos.
What they're called. What are these? What do you call them on sea pens? The like hair-like along the knuckle stalk there. Here, like right here.
Rakis.
What are they called?
Rakis.
Rakis. You see, like this one, the head where the polyps are coming out is really just like a branching point. It doesn't have that same large structure that the last one was going to have. This one also really is large. Translucent. Also has the additional kind of yellow kind of like hairs running across the stalk. para esta solamente vamos a tener algunas vistas, no un pólipo completo porque
Está pequeñita.
Está muy pequeñita y mejor para que no lo conozca.
Se focus.
Yeah, that's a tough one.
Okay. All right, push in Irish.
So you see the base on that one was smooth, didn't have that large ring-like structure on it. Awesome, thank you. Okay. Alright, transecting again. How far is it to waypoint 12? Let me get an estimate there so I can see how. I don't have a. I don't have a good estimate of.
360 meters.
360 meters? Okay, thank you. Alright, we'll go back to transecting and for a while. So we're at waypoint 11. This is fauna sampling and rock collection zone, so. Alright. A line of sea cucumbers. And scallops. So here's some beautiful cup corals on this rock. That sea cucumber has a nice spot of orange. We're seeing these brittle stars laying on the sediment as well. A lot of them, like their main central disc is raised up. It's something, behavior we've been seeing a lot in this area. So yeah, I think, I'm not sure, but maybe after today we're moving to a slightly different location. In the general area, we've been doing a lot of our work in this one bay near the Rhone entrance.
Bridge, RV, new waypoint.
New waypoint, thank you.
But you'll see every time we see a rock, we're seeing some type of coral on it. A lot of times it's either these cup corals or we've seen the mushroom corals as well. I was commenting the other day that I wasn't seeing too many of these small urchins. I was seeing mostly large ones, same thing with the scallops, but here we're seeing a number of what appears to be young sea urchins, quite small compared to the larger ones we've seen of what I believe might be the same species that likes to put little pieces of shell on top of them. It's a nice crinoid, nice peachy colored crinoid. There's a primnoid like the one we collected not too long ago on this rock. We also have sponges on these rocks as well, and a number of different ones. Can we give a quick zoom on that crinoid? Yeah, there's a couple of that coral over there that we recently collected. Amazing. Yeah. Yeah, sounds good. Can we do a Niskin bottle here?
Oh, sure.
Thank you.
Wow, listening to white air.
Awesome, thank you. See, yeah. There's another one of those small, kind of translucent amorulas. Yeah, so we're just continuing to move upslope here. We're on this nice feature where we're going to be going kind of upslope, and then it looks like we're going to, instead of going along, continuing along the ridge, at least for a while, we're going to dip down into a little valley near the summit of this feature before we cross over to a ridge further on in the dive.
How long are we staying in the water here?
Last time I saw, we were going to be on deck at 9 p.m. So we have time. We have waypoint 14 is roughly where they want to do the push cores. Can we zoom in on that coral, on the cup coral on the right? Lower right. Yeah, it's a really long transect. I don't have distances to mark between the waypoints. I don't.
Whoa, that's a huge crinoid.
Coming up. Oh yeah, we can glamour shot that when we get to it. Let's get a nice.
So those transition shots out as well. So that good.
Yeah, well, that'd be more like, I think you want to get there, get in focus, turn off the lights, and
Yeah, and it's a pretty interesting sponge underneath it too. All right. Perfect. All right. I think we're good there. Nice Ian, thank you. Yeah, there's a interesting group of sponges coming up on these rocks. So we were just kind of having a side conversation on as we were zooming on that cup coral. It looks like it is wearing one crazy hat. That sponge. Alex? No? No. Oh, I thought that was Alex coming in again. Yeah, just get the... You can get the lasers on the crinoid, then you can turn them off and do your shots. Oh, and an octopus too. Look at that. Like, there's all these different sponges, brittle stuff, cup coral back there. Octo's coming in. It's like, don't leave me behind. Fish are like, don't eat me. Oh, this is like one of the first octopus I've seen not sleeping. Oh, he's on there. You should, yeah, I don't know, you should. Are we gonna watch a meal? No.
Tienen la capacidad de ponerse como porosidades en la roca mimetizar, y como estaba cerca de eso, quizás intentó parecerse a la piedra.
Brachiopod's like, don't eat me. Ooh, a little color change there. Brittle stars. Yeah, it's amazing.
Yeah,
It's like, no.
Darn it.
Like, nope, I'm gonna sit right here. And we're just gonna sit here and watch for a little bit longer. Get some nice film of a octopus that's not sleeping. Look at the texture around the eyes there. The brittle stars. There's crustaceans in the foreground. Tube worm. Some hydrozoan. Wow, look at that texture. Just doing some. Oh, look at that crustacean on the move. Like, here I am. Don't forget me. I'm here too. I feel it's like from that Star Trek movie at the beginning, like, oh, volcano. Star Trek. Mm-hmm. No. Oh, was Patricia just in here?
Yeah, she was here. She just left.
Oh, yeah, I didn't see anyone behind.
No, I wanted to
Thanks.
confirm that she won.
Okay. Yeah, because I think they're going to do a zoom on the crinoid still with the sponge before we're trying to wake it up. Okay. No, hardcore sleep. Passed out. It moved for a few minutes. It's all right. Can we just get the if you want the glamour shots here on the sponge and the crinoid, and then we'll want to collect the isopods that we were just zoomed in on with suction. Not to collect, no, just a glamour shot, whatever glamour you wanted to do. That was our original pause, and then yeah, and then you guys saw the octopus, and yeah.
You're no longer fine. Some sideshow ball. Yeah.
That's funny. It really does look like a Muppet. Yeah, the isopods that we zoomed in on at the end there for the glamour shot. No, they are. I think it was more the mix of the cartoon appeal of an enormous crinoid on the sponge with a mouth. Yeah, awesome. Okay, so yeah, back over where the octopus was. Yep. So right, these two right here. We wanna. Yeah. Yes. The stow shield. Yeah. The show stealers. Ooh, there's a little hermit crab, I think, on that one. All right. So it kind of reminds me more of like beaker.
Yeah, he's very beakery too.
He's very beakery, just his dimensions are off a little.
Alright, yeah. Keep your camera and just give it a focus. Suction.
Alright.
What is the chumper? What's chumper?
We should probably move suction over to, these are pretty big. So which one are we on now? 6?
6. Yeah,
Maybe let's go to 4.
4. Okay.
Is there something in 6 somewhere?
Yeah. Taken over.
Okay.
And 5.
4.
4. Alright, so
Okay.
Let's see how many.
Yeah, there's a total of 3 of them.
We're in the good jar.
Yep.
Alright, selection coming up for blast.
They're holding on. Yeah, we're probably gonna have to take some of that with it.
Yeah.
Oh. And a sipunculid or a tube worm. And there's actually one more in there. We have to try to snag that 3rd one.
Oh, I see.
Oh, you're going for the triple kill, are you?
Yeah, we want to kill all of them.
Jeez.
We're 3.
Yeah. For a lot of groups, you need 3 individuals to.
3 now.
Yeah. To describe species, do whatever. I have no clue if these could be a new species or what.
Okay. Octopus friend is not unharmed.
All right.
These 2 guys.
Point out.
There is this 3rd bundle of tumbleweeds.
All right. Perfect. Thank you. All right.
One's jar 5.
And coral and So is there? I think there's something in 5. What does it say? Polychaete.
5
Let's
say
not.
polychaete.
We should index to 3.
3.
Yeah.
6.
Yeah, I have something in 5, and they have something in 6. Do you have something
Yeah.
in 6?
No.
You don't?
No? The pycnogony? The sea spider?
No, I didn't see anything in 6 either.
So we got 4, 5, and 6 closed now. Okay.
Yeah.
Maybe.
Cool.
Yeah, 4, 5, and 6. Isopod, polychaete, and pycnogony.
Okay.
So anything else you want to do in this little wonderland?
No, we can go ahead and continue transecting, make some progress. Like, Aaron likes the sample. Okay. Onwards we go.
Not too much.
Yeah.
Twice in a day, no.
Oh no, we're a ways away. It took us an hour and something to go. So we should at least try to make it to the next waypoint by the end of the shift. They had a 2-hour time window from like 9 to 12. I don't know how long they were in 9 and 10. I started in between 10 and 11, so. Lasers on. Thank you. So yeah, just try to do a little bit of transecting here so we make some good progress. The sea urchins that we often see with a little piece of shell on them. Sometimes a large piece of shell, like that one on the left. Still a pretty impressive sea cucumber field here. So yeah, for anyone just joining, we are currently exploring an area of seafloor that underlies part of an ice shelf that recently calved off and is exposed for the first time. The overlying ice shelf is kind of like part of it has come and gone over the last couple decades, but the part that we're exploring is the part that has never been exposed in modern times. So as the Earth continues to warm and warm waters are coming in particular in this area of Antarctica, we're seeing accelerated warming and melting of your ice shelves. And they're being warmed kind of from underneath as the water flows underneath the ice and helps thin it from below. So you have these large calving events forming these large icebergs. So that happened just when we were in the area about a week or so ago, and so we decided to come over and explore this area that not only has never been explored because few expeditions have come into this basin, but because it was actually covered up by ice for centuries. And it has just been exposed for the first time. We've been seeing a lot of sediment on this dive, but in this part here, we're starting to see a bit more rocks. Every time we see rocks, we're seeing hydrozoans, cup corals, other groups of corals as well. We have brachiopods, some sponges on these exposed rocks. A lot of filter feeders. There's a tunicate there on the left, the kind of gelatinous looking. And mixed amongst all of them are some brittle stars, isopods, amphipods. We've got some sea urchins. We've seen some nice crinoids as well. Is that a large sponge or is that a rock?
Pretty spongy. Check it out.
Yeah, let's just give it a quick pass by with the lasers on it. And there's an octopus by it too. Mm-hmm. That's a nice view. There's an anemone over there. Ooh, look at that brittle star.
There's a bunch of your isopods on that sponge.
Mm-hmm. Yeah, a little isopod party. That star looks like it's all wrapped around itself. Ooh. That was a scallop. I don't know, I'll let Alex decide if he needs something. I think it's a little sedimented here. Cool. I'm good. I think we can transect. It's just kind of sedimented here, so I don't know. I think it was more appealing with the other stuff around the other one. Yeah, so we can go ahead and continue transecting. We got the lasers on that other. Is it going down slope now?
I was coming up, so when I saw
You're coming up, okay.
But
We're confusing because, oh no, it's right, okay. Just getting confused at which waypoint we were at for a minute, and I thought the two screens differed, but they're good. My brain is just tired. Okay. Alright. Back to transect mode. So yeah, we're amongst these fields of sea cucumbers that we've been seeing, and then rocks with sponges and corals and other organisms on it. We're also seeing the occasional sea pen. It's been pretty diverse, I think, in terms of what the habitat looks like. We're seeing these lots of rocks intermixed amongst the sediment. So a lot of these might be maybe some drop stones, some exposed bedrock. Trying to look for an interesting, there's a nice crinoid swimming. You can turn the lasers off if you want. See, we didn't have to disturb the crinoid earlier. We got one that's swimming for us. Oh, coming in for a landing. I've swum enough. Let's see where I get. It's amazing. So pretty. Purple and orange, like two of my favorite colors. Oh, nice. Yeah, the little urchins back behind. One's wearing a nice size. Fabulous. Thanks, Ben.
It's amazing. Wendy.
Almost. Alright, pick it up. There's that rock there.
Okay. Oh, that's a nice rock. Are we gonna, let's sit down and collect that one. We're due for a rock collection.
Due for a rock.
Seems a nice small rock.
Oh, wait. Are you in Berlin? In Berlin?
Yeah, there's two of them. Yeah, there's two of them, and I went ahead and stacked them because I don't think we're gonna sample much from the one. I think it's just gonna go to the museum.
Okay.
Or we'll just take like a little piece of the tentacle, like a really small amount. It's small and kind of. Yeah, 56 meters. Once you have it, we'll bring it up for a quick zoom and then put it in rock box 1. That's out on the arm. Going on the arm. Yeah, that's a good side to view, I think.
Some quartz.
You see some of the beautiful rocks we collected yesterday or overnight, Zach?
I saw
Some nice
out there,
pink
but
ones.
I think I need to go look. You said pink ones?
Yeah.
No, I didn't see those.
Yeah, like the last 2 nights we've collected one on our on our shift. Awesome, thank you.
Alright, so you want this over on the side?
On the side arm, yeah.
Is that rock basket one?
Yes, rock basket one. Perfect, thank you. Alright, transect mode. Perfect, thank you. Up and away, back to transecting. Still trying to make it to waypoint 12, but we've seen some fauna that we haven't seen before. So it's been pretty interesting. This is shallower than I think any of our dives have been. So that might be why we're seeing some of these different species, at least for the sea pens. We would need to do some more transects to, I think, confirm that, to see in multiple places they only occur or show up at certain depths. Here we see a number of mushroom corals, some small one on the right, and this larger one with the polyps kind of partially retracted on the left. It's a nice size anemone in the distance there. Yeah. Mm-hmm. Yeah, this is Anthomastus, or a closely related group of corals. Yeah, we're kind of going up this ridge right now, and as we get to our next waypoint, we should be in a small valley in the ridge. We'll kind of be going like up and down a little bit for the next couple waypoints. Seafloor here has a lot of like up and downs, a lot of complexity in the features. So since we're doing a little downhill here, we're just like reversing down the slope. It's safer for the ROV. You can see where they're going better. A nice size of anemone here. A nice sponge on the right. Yeah, you can see a lot of these rocks have these mushroom corals on it. And sponges. Yeah.
Yeah, we did that the other day, man. I followed this for a long time.
Who did we send sliding? Just a rock?
Just a really unlucky person.
Nice sea pen. Just working our way backwards down the slope. There go the sea cucumbers. sea pens are pretty. No, we have quite the density of sea cucumbers here again. It's been amazing on our shift so far. We've seen these sea cucumbers pretty much consistently, and we'll hit these fields that are really dense. There's a different species mixed. We're now seeing this other species of sea cucumber that I haven't really noticed before, but all of a sudden they're starting to be mixed in. Quite large. We might have seen them on other dives, just not on this dive. I hadn't noticed them. Okay, we've kind of hit this flat area, so we're going to be able to turn the ROV back around into the orientation that we're heading.
He's got a Oompa Loompa stuck on his front bumper there on the left.
Yeah.
See it every once in a while.
That's not this one. Or is it still there?
I think it's backed up. See it?
We'll see how long it stays there. That's funny. Oh, here.
I hope that's not the one from our bottle box.
No. I don't. Maybe. I don't know. Maybe. That would be funny. I don't know how it would have escaped, but maybe. Yeah, we should probably, when you sit down, check.
Yeah, we'll check on here.
Just take your porch out, we should be able to see.
Yeah, that's it. It somehow made an escape.
Is it?
Yeah,
No
I think
way.
so.
It wasn't.
No, nope, it's still there. It's a different one. Okay. I thought. Yeah.
Sea pen escaping.
I was like, how did that happen? Because we had a starfish escape on our last expedition, and I watched the video, and that starfish moved from one part of the bio box, like it was in a quad, and it moved, like it passed through all 4 sections of the bio box, and then like in our next view of it, it was just like gone, and it was never seen again. And so there's apparently a spot in the back of the box where there's a really small opening, and so it must have made its way out through it.
No way.
Like up near the hinge or something. So yeah, it was on the move. It went and visited the other starfish and the other part of the quad. And yeah, I put together a little time series of snaps looking down at it. It was amazing.
We got a real cooler now on this up if we ever need to.
Oh, nice. Yeah, that's something. Maybe, I don't know if they'll want it on the next expedition, but there was someone who was going to come on this expedition that maybe would have wanted that, but. You have to bring it up for the next cruise. Or I guess you're not going to be here at the. Pass it on to Doug. Again, I don't have to make any decisions on that one. I'm just told what to do. Relax. help process samples, leave the decisions to other people, give my feedback. Nice. Yeah, wow, so many sea cucumbers. Yeah.
It's weird how different it is.
Yeah.
Where's that person that wanted these kind of sea cucumbers?
We collected some earlier. I think she wants some more from shallower, so I haven't heard of. Yeah.
When she collects, it's good to plant into the tree and
Yeah, she can.
Got a couple of on the board.
Everything. Wrapped around those rams.
Ooh, big
Sponge.
sponge. Looks like a snowman, his head's in front.
Kind of looks like the sad snowman that, like day 3 of the snowman that was on the ship. A little melty.
Do you look like Borderlands?
Incredible.
My name is Mr. Tori Tubbs, like a professional wrestler, and that's, I can't not read that in that.
Wow, this is gonna be fun for someone to count.
Someone to count, huh?
Mm.
Oh yes.
So many sea pens.
We can spend the entire week.
Looks like a exposed rock or a giant. No, it looks like a giant Dumbo. Oh my gosh. Yeah, we do. Yeah, get the lasers on it, and then it's an enormous Dumbo. I thought it was like. Yeah, just get the laser. Yeah, I think as they're sitting down, they can get it. Mm-hmm. Wow. Alright, let's grab some. If you pop the lasers back on just for a second, they were almost on it. Oh, okay. I see. It was just the camera was moved too far. Push cores. All those push cores obstructing the lasers.
There we go. All right. Yeah.
See like the area it cleared before it pulled in all its tentacles to tuck up to sleep.
I'm thinking like how a dog like has a circle and then
Mm-hmm.
Alright, so you're good with lasers out now?
If you can just pan over to see if you can get the lasers on it. Pan up, I don't know. I guess it's a little far.
Are you racked out all the way?
Should be.
Alright, no, they're just being obstructed. That's alright, you can go ahead and turn them off. And then we'll just grab the lasers on it as we're flying away.
Should I give it a little boot? Because I kind of want to see that turn into an octopus.
No, I don't think we should. I don't think it seemed to work very well last time. It just ignored. It was like, I'm asleep.
Moving going.
It's like. It does look pretty cool though, the way the sediment too, like settled on it. And all its little creases and wrinkles. Aww. Friends. Like, this is a good place to sleep.
Yeah, that seems safe.
Yeah.
This guy's doing it.
They have the same color.
Yeah. They do match. Nice purple. I think I was kind of sleeping against it. I think you're so soft. You're like kind of bouncy.
Holding on to it. Maybe that's just pressing its face.
I think it's just pressing its face against it. Like its chin.
Best friends.
Yeah.
Yes.
That's cute. All right, well. Buoyancy problem.
Well,
Yeah, onwards. Yeah, going to continue. See. Let's just see if we can get the lasers on it so we can get a nice size estimate of the Dumbo.
there.
They're good friends.
Yes.
Yes, very good friend. Alright, in transect mode.
Laser this guy.
Oh yeah, laser him, but yeah, it's in transect mode. Sorry, I got distracted.
Switches out just the hair. Lasers.
Awesome, thank you.
Doing been lased. Shoot. Big bulbs, eh? See you later.
Onward we go, transecting to our next waypoint. There's quite a distance between these two waypoints, so it's weird. It looks like we're past them on that way that map is.
That is weird. Flare mouse, since that's where beyond 12. Somebody's been knocking me somewhere.
Yeah, just the height thing again. Yeah.
Complicated, it's probably.
Sea pens, fauna forward floating by. We can just keep going. We should play a little catch up before the next shift comes on. I know, but what can we do? There was an enormous Dumbo octopus.
Yeah.
Like,
Right. You gotta stop.
yeah, you can't. Yeah, can't not stop. mean, sea cucumbers are cute and all, but yeah, I think that's like the faux tripod fish that we've been seeing. So I think. Yep. We can go and keep going. Yeah. The glowing. I think they actually collected one of those fish the other day too. Just, I think Tom shared a photo of it.
How's the squid camp then? Haven't checked in with Tom on that.
I haven't heard anything yet. I'm assuming we would have heard if something happened. Have you heard anything, Aaron?
He has captured some squid.
Really?
In it. Yeah, there was one that came and aggressively came towards it and like.
Colossal squid?
No, just, I think regular kind of squid that we've been seeing, like the ones that I think they're similar to the ones that we saw dive bombing into the sediment.
Has he thought about putting some bananas down there for scale? Because it's hard to identify them.
You're going to ask him.
Necessary.
What? This one? I know.
Spawn.
Yeah, a little sponge field. But I feel like mission is accomplished in this shift. We got some samples and got some nice footage. Yeah, these are some nice size sea pins. Yeah, so these are more like the sea pins that we were seeing on the other dives and different from the ones we collected earlier today. Yeah, it's sea cucumber paradise.
Oh, definitely.
We're still on the search for some rose-colored rocks as well. Those have been particularly interesting to the geologists. They appear to be more volcanic, I believe, in origin. So they're different from the majority of the rocks that they've been collecting. Gives them a little more complete history and our understanding of Antarctica. Nice rock outcrop. It's interesting, we're seeing kind of this like rosish colored sediment now.
I think it's moved by the seabed.
Oh, it's pretty purple and orange.
Mm-hmm. Yeah, these crinoids are really pretty.
Very unique colors.
Mm-hmm.
You don't see that in nature much.
No. It's a pretty fabulous color scheme. I think Sebastian could benefit from some purple and orange.
Different colors.
Yeah.
Are you
You guys for a while had some purple on Sebastian. It was very pleasing. No, that would be amazing. I wonder if this rose colored is coming from the urchins. Curious.
There's some like there's that red dirt almost on top of stuff.
Mm-hmm. Nice sized rock.
Just one side of it.
Mm-hmm. The favorable side, yeah, currents more favorable on this side.
Bridge, ROV, new waypoint.
Yeah, so it's interesting. You can do a circle around a rock, and one or two sides will be loaded with other organisms on it, and then the others might be sediment covered or mostly bare, depending on its orientation to the currents. And this side is the ski slope. Yep. Yeah, they have on here to push core around waypoint 14 is the target. They have a target to push core around waypoint 14, which I think is near the shallowest point.
How shallow is that?
To be honest, it's right around the depth that we're at now, but I think a lot of their depths on here for their waypoints are different. Like they haven't been very consistent with what the depth is when we actually reach a waypoint. So now it says we're supposed to be at 395 meters, or when we hit waypoint 12, and we're going to be shallower than that, so
Yeah, 30 meters.
I think, unless there's a sudden dip down at waypoint 12, but it doesn't look like it on on Flighter Mouse, so we're gonna make it to waypoint 12. This is a long trip. This is Rhynchoceranthid, yeah.
Let me guess, you wanted to grab this one too?
No. No, this is not a sea pen.
And that is
not a sea
weird.
pen.
No.
What the world is that?
It's like,
I bet if you came around this way and looked right in there real smooth, it'd be cool.
yeah.
But look at the proportion with this sea pen. It's huge.
Yeah, those are pretty small sea pens though. That's close to 5 centimeters, just like it's...
But look at this. Really amazing.
That is
Look at the colors.
Yeah, you can take the lasers off if you want.
It's like taller than a human. That's great.
Alex.
Here it is.
Yeah, cool or what? Ooh. That's the same thing. That's from where?
Oh, nice.
Lasers are off.
Yep. Yeah, I was ahead of you on that.
Close it a little? How's that?
It's really nice.
Yeah, did that lighting?
I'm lost.
Turn off the light, kid.
Yeah. You got it down right now.
Yeah, Rhynchoceranthus. Or Ceranthus, not Ceria. Ceranthus. Okay. Yep. Don't let me forget.
Okay.
Unless you want to use the center cap, that's
Ooh, isopod.
Oh, it's very tiny.
It's trying so hard. Oh, amazing.
ROV bridge. I think we have to do a little step to port side to avoid a little bit of ice coming for the umbilical before continuing our move.
Roger that, Phil.
Hey, Father.
Yeah, again, I don't have any information to give you an age estimate on this one. I don't know if work's been done. But yeah, I would say, compared to any of the others we've seen, quite old. It's notably larger than the other ones that we've seen so far on this dive during my shift. Mm. So Maxie is telling me it's a Corymorpha, the genus, or the Ceranthus.
Wow.
We're also getting ready to do shift change here, but a lot of people are coming in to view the beauty, as well as shift change for both the pilots and the science team. Oh wow, that's like glowing. Oh wow. Yeah, maybe someone coming in on next shift could make an educated guess. I would really just be guessing right now, so I don't want to do that. I prefer to give you all some sort of educated guess. Ben just came in for Tyler, and it looks like Joel is going to be coming in for Zach here in a bit, once we're done with the filming probably. Hey Ben.
Hello.
How are you? Thanks for the picture. Okay, I'll send you a bill.
Wendy and Daniel.
We're still doing some filming here. I'm still doing some filming here, so I'm just gonna pass over in a moment. So I did an unusual thing. I put 2 sea pens in here, mostly because I didn't want to use up all the bio boxes, and these were 2 species we hadn't seen before. And I think the one's mostly going to be museum collection, so there are 2 sea pens in there. This one is kind of sticking out, so it's kind of, I don't think it's going to float away, but it was really big. There was some logging issues, and so we think there's a picnic gourd going in there because it was on one of the 3 sheets, but not the other 2. But 4 and 5 have stuff in them. And we've gotten our rock for this waypoint here, so you'll be, I don't think you're due for another waypoint or another rock for sure until 13. We're pretty much at 12 now, so she wants to transect for the instructions uninterrupted largely from 12 to 13, and when you get to 14 is when push core time is supposed to happen, unless it's indicated otherwise. We want to get a Niskin bottle after we finish imaging this, and then ideally it's quite big, so we kind of got the lasers down near the base, but if we could get the lasers before you fly away on the crown, yeah, that would be great, so they can get a better size estimate for the analysis they're going to do.
Do we recognize this?
It's a Coriamorpha, or Corimorpha. Always add extra letters. It's the genus and species names. Yeah, it's a Branchioceranthus, but yeah, it's definitely not worth it collecting. They turn into gelatinous goo when you put them in ethanol. Yeah, and it's huge. So
we get to do the Niskin for EDLA?
yeah, we want to do the Niskin. Yeah, it'll just be the next Niskin bottle here before leaving as soon as they're done filming. So Alex has been coming in. It's been some interesting stuff. So he's been doing some filming for the highlight videos and stuff. So if he just comes in, just you're welcome, Alex. All right, thank you. All right, since Alex is done with that, can we grab a Niskin bottle number 3? At 358 meters. All right, I'll leave you all with the next shift.
Thank you.
We already did that. They already did the whole thing. I don't know if they want another set when they get shallow. I haven't heard, but I guess not. That would be the only ship. We have it in time.
Which species?
I think I didn't get them there before. I think they were the one she usually, the Scrotoplania is the normal one. We haven't seen kind of these purplish, bigger, brighter ones.
Are you Bridge? Do you want to stop here?
At least one of those.
Yes, please, great shots. Stop for a
No
minute.
urgency to get them, but I just need to get representation for one of this. Yeah, it's probably kind of purplish color. We've seen a few when they've been smaller.
Shall we proceed? Before we head off, could we get the laser scale onto the crown? Yes. to yeah, if we could get them onto the crown, onto the pinky bit. Just the rest of it's so skinny we haven't got a.
You just want the crown?
Yeah, just so we can get a proportion. Maybe along this sort of bell shape.
Are you trying to size it with the base?
Yeah, because it's hard. Because so maybe the sort of bell shape of the base. That's it. Yeah, both the same plane.
Yeah.
That's it. That's great.
That's about 20 centimeters long, twice as long right now.
Great, because we just got the sediment and a little bit of the stem, but it's hard to get a proportion of just where it is. Very cool.
I can see it from here. It's quite big actually.
Yeah, you get a sense of scale when you've got the cores in the front.
The laser are 10 centimeters piece, about that far, and down is about twice as long.
Yeah. Stunning. Great.
Hello, my name is Pedro Martinez. I'm a marine biologist from Spain, but working in Germany.
That's it.
At the Senckenberg Research Institute.
Right, and that when it shines through that.
I'm a professor for marine biodiversity at the University of Oldenburg, and I will be here with you for the next 2 hours. And leading the dive is Tom.
Hello everyone, I'm Tom Lindley. I'm a fish person, a curator of fishes at the National Museum of New Zealand, Te Papa Tongarewa. And hello to everyone who's already on the stream. We're just settling in, we're getting a bit of a sense of our bearings, what's been achieved so far, what our next objectives are, and then we're going to make a move.
We just came in and we had this magnificent anemone or polyp. It is almost 1 meter high, I guess. I'm guessing. It is so beautiful. These organisms are filter feeders, so they are capturing the particles from the water. And as we can see, there is also a lot of particles in the water. So it seems to be enough food in this environment. We are now at 358 meters depth. I think that's the shallowest location that we have sampled so far in our Antarctic trip.
Until the next waypoint, it's a video transect, so the usual 1 meter up, 1.3 speed, lasers down. I'm going to try not to get distracted and see anything exciting. But you know it'll happen.
Which waypoint are we at the moment?
We are right on 12, and they've requested a fairly uninterrupted transect until waypoint 13, when we're going to do some more collecting.
Yes, I wonder if we should take the bush corals before that.
Do you want to do that?
Yeah, because then it's getting more scarf, so this is a good flat area here.
Okay.
And then
I've got the preferred push cores at 14.
No, but this is a hole.
Okay.
So I think before we proceed with the video transect, we should then take the push corals.
Okay, come down to a stop.
And before we take the push corals,
cool.
Thank you, because it just did one, so it's fine.
Are we taking all the push cores?
Yes.
Yeah, there should be 17 left over.
Right. Okay.
This area is also full of sea cucumbers. Oh, now we see some outcrops here. So, of course, for the push corals we need sediment.
We're feet close to a rock. Do you want to pick one up close to a rock, or do you want to?
No, it's better not so close to the rock.
From that
So we're landing now.
Okay, we've got quite a shopping list of push cores. We have 12 regular sized ones with just 2 centimeters of water in the surface so that the surface of the sediment is undisturbed. And then we have 3 long and 1 regular one all the way to the top for geology.
Are we taking all of them?
Yes.
It's looking that way, yes.
That's really taking. Oh no. I'll take it.
With them all.
I'm gonna porch in just a little bit so it's easier for you to see it. So we're gonna start from the front.
It's a little bit gravelly here. So if we take the geology ones first, so
That's what we're doing. This is all geology. The front ones, I think.
no problem, no problem. So these will push all the way in, so the sediment's right up against the bunk.
You see that the push cores, they have numbers, so we can identify them later on. Here we are taking all of them at the same place, but we could also choose to make a transect and take whatever 2 push cores every half a mile or something like this. So it's important that we can recognize them later on. We record the
We do want all the long ones as well.
coordinates and the depth.
Yes, I believe so. 17 remaining. Some of these coras are a little bit longer than the others. I think they're an extra 10 centimeters maybe. Trying to remember. I think the smaller ones are 30 and the larger ones are 40. And they're indicated by these rather fetching stripes down the side. Little mysid coming in for a look. Right now we're taking push cores. Simple bit of kit, but incredibly useful. A lot of the teams are after these. So you can see that spring-loaded cap at the top. It's a one-way valve. So as we push it into the sediment, it allows the water out the top, and then when we pull these out again, that one-way valve means that we pull a plug of sediment out with us. And they do a really good job, actually. It's really undisturbed. It doesn't tend to lose much material. It just takes a steady hand with the manip.
Sediments inside the push cores will be used for different disciplines. For the geology, to look at the different stratigraphy. We will look at the chemistry of the sediments. There are some people looking at foraminifera, that are organisms that leave also a fossil record. So they have shells that are retained in the sediment. And this can be used also, the chemistry of the shells, to reconstruct the temperature and oxygen in the water in the past. Other people are looking at the meiofauna, so these are very small organisms like coppepods, crustaceans, or nematode worms.
That was another long one.
All of them smaller than 1 millimeter, living in the sediment. And we want to record the abundance, the biomass, and the biodiversity of these very small creatures. Other people are looking at larger animals in the cores, like polychaete worms or bivalves, small crustaceans. So therefore we need to take a big number of cores to have enough samples for the different disciplines. For the geochemistry, the total organic carbon is measured, the amount of chlorophyll in the sediment. This is an indication of how much food is coming from the water column as an energy source for the benthic animals.
Final long one is 3. Is 3? No, 3 should be a gap. As they said, the geology. Yes, yeah. So this one will be all the way to the top again, and then we'll just do the others.
I think you want, I can punch in too. You want to grab the side of it. Okay, it's up to you. I'll punch in, see what happens. Okay, you want to try to grab it?
Don't worry about it.
I'm watching.
Okay.
You want to try the other one?
Yeah, that's right.
We've got a few different camera angles here in the control room. It can be difficult to figure out just where everything is in space and to coordinate the manipulator arms moving over this tool tray. So we flipped to a few different views just to make sure what plane everything is in. You're not going to contact anything else. The arms are incredibly strong. These squidgy looking bungs that they're grabbing are really difficult to squeeze with your own hand. So the arms are incredibly strong. So have to be careful we don't damage any of our own equipment. There's also a sort of range of movement to each of the manipulator arms and a bit of a division of labour between the two arms. So this is the one that can more easily reach the left side, but it's the more gentle of the grips. It's got these yellow pads on it that make it a bit more gentle. It's got a cutter that allow us to prune things like corals.
You're good. Watch. You're still good to go. Or just do it in front.
is a lot of shells on the surface of the sediment. I think there are shells. It could also be little gravels or pebbles.
I've seen a little bit of both. It does seem different here than last night. There's a sort of shell hash and gravel here.
I don't know if it'll push straight. You can just keep going. No, I'm watching it.
All the way, you know?
Okay, Mark. Perfect.
Here we see on the top right this fantastic umbellula. This is coral. You see the different polyps at the top. This particular species is relatively common here. We have seen in different dives, and they can also get very large, like more than 1 meter high.
So that completes our geology once.
Okay, I'm gonna porch out again. Okay, I'm gonna stop it here, and then we'll just porch out as necessary. Still good.
The oxygen concentration here is indicated by 53%. This is a little bit higher than what we had in the deeper areas. In the deep sea we had about 51%.
Our biology, so just a couple of centimeters of water left.
In general, the oxygen concentration here in these Antarctic waters is very high.
As long as there's a little gap of water.
We have not found any sign of lack of oxygen in the sediments. The waters are very cold. We have now, I think, 1 degree, or it's indicated 0.7 degrees. And well oxygenated waters.
Somewhere where you can see it. In your view.
And that's the reason why the deep sea waters in all oceans are cold and well oxygenated, because the Antarctic shelf is the origin of most of the deep sea water that we have in the oceans. When the water gets cold, it gets more dense. Oh, here we have a fish saying hello to us. So when the water gets cold, it gets more dense, and then it falls to the bottom, and through the continental slope, this cold and dense water flows into the deeper areas of the different oceans. The Antarctica as being in the South Pole, it has contribution to the Atlantic, the Pacific, and the Indian Ocean. So this is the major contribution to the deep water formation on Earth, and the start of a deep water circulation that take 1,000 to 1,500 years until these waters come back to the surface in upwelling regions. This is the great deep water conveyor belt.
A couple of times in Earth's history, that conveyor belt has broken down, and the large ocean basins have become stratified, have become sort of blocked off deeper and upper, and they're associated with massive die-offs of the deep sea. So a few times in Earth's history, the deep sea has had a big reset, and most of the life there has been lost. And those sort of conditions are what's created things like our oil deposits. So everything died off very, very rapidly. There wasn't enough oxygen for it to be broken down, and then it got trapped within the sediment and chemically altered within the earth rather than broken down by bacteria and things like that. So that is one of the Earth's mechanisms, but that's what we're worried about triggering again.
The quality of these cameras on this ROV is fantastic. It's one of the best, I think it's the best quality that I've experienced in different ROVs. It's high resolution camera, 4K. And the photos, we are getting a still photo every 10 seconds. They are also of amazing quality. I think one of the success of these images is that the illumination is also very good. Some of the ROVs that I have worked with, they lack a little bit less light, and then of course the images get not so good.
Yeah, it's more than just raw resolution. You need enough light to drive that and some good optics. And this is a really nice setup.
This camera has also very nice zoom, that you get excellent close-ups. For the people that just started watching our chat, we are in the Bellinghausen Sea in Antarctica. This is a place western to the Antarctic Peninsula. It's a place that has not been visited so often by scientists. Other places around the Antarctica, we have much more information, like the Weddell Sea. Many stations are located in the Antarctic Peninsula and also in the Weddell Sea. And specifically, the dive that we are having today is in an area that was covered by the continental ice for thousands and thousands of years. And only recently, this ice broke and started moving out of the continental shelf with the currents. And this area got free of ice only a couple of weeks ago. So it was not planned by our expedition because we didn't know that this ice will break. The ice shield that broke is about the size of Great London, about 30 kilometers by 20, something like that. So it's a big area. And we had a great opportunity to take samples in this area so closely, so short after it has been freed from the ice. Our expectation was that we will find not so many organisms at the bottom, because when the water is covered by ice, and the ice shield is about 150 to 200 meters massive. There's no light coming through, and the algae, they cannot do photosynthesis, they cannot produce biomass. And these communities at the seafloor, they need the energy coming from the water column, from the water surface, in order to survive. This is the food that they get, the primary energy source. And to our surprise, this is the third dive that we are doing here. There is a lot of life, there is many organisms, and also a lot of different type of species. The biodiversity is quite high here. This means that this area also was covered by ice. The ice was not reaching until the bottom, because in some areas the depth that we have sampled was 1,200 meters, so the ice is only the top 200 meters, and then we had about 800 meters of water column. So it seems that this area was still connected to adjacent areas, nearby areas, by water currents. And through lateral transport, these water currents were bringing also food to the communities living here. We have seen in the dives, when we disturb a little bit the sediment with the ROV, some areas were very calm, so the sediment took a while until we got a nice free view again. But in other areas we saw that there were very clearly bottom currents of several centimeters per second. At the moment we don't know exactly where these currents originate or where they're coming from. It might be that they are tidal currents. Also here in Antarctica you have a tidal influence of the tides. And this is something that we will analyze later.
These little mysids are enjoying the light. As we stay here longer, we're gathering a larger and larger crowd of these little shrimpy looking guys. We might end up with a free mysid if we're not careful. Ooh. Oh, close. So we've noticed some organisms are attracted to the lights of the ROV, and some seem to be spooked and driven off by it. So if we set up shop for too long in one place, we end up with quite the following of the things that like it. I can see on the rear view camera there's quite a crowd as well. When I was doing some work on the mid-Atlantic ridge, and there was one particular type of fish who'd quite liked the ROV, and as we did a longer and longer transect, we'd have more and more of a crowd following us behind, like ducklings. So it's funny to look in the rear camera and see just this gaggle of fans following the ROV.
There was a question in the chat of how many people are involved now in this, what we are doing. We have 2 ROV pilots. One pilot is more devoted to control the ROV, and the other pilot is in charge of controlling the arm, the manipulator. And we have 2 scientists giving the instructions and 2 other scientists logging the samples, taking photos, logging the coordinates, taking notes of what is going on. And we have also a social media manager taking care of the 3 streams that we are getting a live stream on YouTube, Facebook and Twitch. And in general, on the vessel, we are about 26 scientists, I think we are.
I think so.
And I'm not sure how many crew, but I think there are more, like 40 crew, something like this.
I think we're just over 60 for total vessel complement. Then, of course, there's lots of shoreside support as well. So depending how far back you want to radiate, there's a lot of people involved. The manipulator arms are controlled with a sort of proxy for them essentially, so that you can get a sense of the range of movement and where each of the joints locks out. It is a miniature version of the arm essentially that has a one-to-one translation to the arm you can see ahead of you. And so by moving this scale model essentially, your movements are translated, and you can adjust the sensitivity a little bit so you can do sort of broad movements or finer control. And we were talking last night with the pilots, and there's not really a perceivable lag. We're connected by a fiber optic cable, and so the translation is almost instantaneous. There's probably a larger lag actually by by translating that to hydraulic movement, then there is a lag in the signal.
Yeah, it is very nice to see, very interesting to see that the movements, there's a joystick, and the movements done here on the control room, they are translated one-to-one to the manipulator arm. I read the other day that today is also possible for doctors to make some surgeries kind of remotely, sitting on another hospital and using manipulator arms on other remote places.
The arms are not identical, so the one that we tend to use here is a bit larger and stronger and is more about gripping tools. You'll see some interesting shapes within the claw, and they're not random. We've got some gripping pads that meet, but you'll see there's actually some channels and a cross channel as well. So that perfectly relates to the tools and the handles on them. It's usually called a T-grip. So when we pick up our tools, if we've got a good grip on them, they lock into that claw and just become an extension of the arm. We also have a camera on the arm. You can see here the light's on, and then there's a second camera just on the wrist joint, and that gives us some nice up-close view of what the arm's doing. On the other side, it tends to have a longer sort of pincer. It's a little bit gentler, it's got a soft grip, and that also has a cutting blade in that pincer as well that allows us to sort of take, like pruning scissors, take some cuttings off things like corals, so we don't have to tear or pull pieces off, which can damage or uproot a large colony. We can actually take a little snip and do minimal damage, but we still get our sample.
Seems to me that there's shells right at the bottom.
Yeah,
Little shells.
They look like bivalves. I see caps in them.
We have had in other dives a lot of scallops, also some large ones. But these look like other type of bivalves. Look like
a bit more clam-like.
Clams, yeah. The depth rate of this ROV, I think it's 6,000 meters, but
4,500.
4,500. We got the information from the pilots.
There are a couple of full ocean depth ROVs. Some are coming back online now. The previous generation, I think we were up to 4 at one point. They sort of wore out or were lost or damaged. Some of the other models, they kind of don't exist as a singular vehicle. They exist as a pool of equipment. So, cameras and manipulators and things like that are sort of swapped onto other vehicles. There's a few different major types, different major launch types. So we have an umbilical with some floats on it, which is coming out the back and pretty much going straight up. Some of the deeper systems, and maybe it's phasing out now actually, but they used to have sort of a launch vehicle, a launch bay, so that would go straight down, and then the ROV would launch out of this vehicle on a spool and sort of do horizontal transects that way, and that made things a lot more complicated because it did have thrusters on as well, so that became an extra vehicle you had to coordinate, but it did mean that your umbilical was then going horizontally, and in some situations that made movement a little bit easier. We have this very strong armoured cable that allows us to haul the ROV up, but it's also driving the voltage down. It's direct current, so you get voltage drop over long cables, and so you've really got to push a lot of voltage at the surface to get enough to the vehicle. Some of them got around that by taking their own internal power, so you've got ROVs with internal battery packs, and then you just needed communication, and some of those adopted just the fiber optic tether itself, and it was staggering. It was a whole ROV system. This was the Nereus system that I got to use in the Gamadic Trench. It was like a human hair. It was hard to see. It was like a little bit of fishing line, and that was all the comms coming down this fiber optic cable. And it's the same as the torpedoes used. So actually, you can steer and control torpedoes because they spool out on one of these fiber optic tethers. It's great for transects. It's great for when you're sort of traveling along, but it's very easy to accidentally sever it when you sort of make maneuvers. So we lost comms quite a few times.
The mean depth of the oceans is about 3,500, 3,600 meters. So with this ROV you can cover probably almost 90% of the oceans. Then there are some areas which are very deep. These are the trenches, but this is only, I think, 2 or 3%. Is this right, Tom?
Yeah, it's about 1.5%.
Yeah.
So it's 45% of the total ocean depth, but it's only about 1.5% of its sort of surface volume. So the Hadal trenches are really interesting because they go super deep, places like the Mariana Trench, but they're actually quite rare and quite spaced out. So you get almost island biology there. You get things that are only found in their own particular trench. Once you adapt to going that deep, it's hard to get out again. So that was core 17 for biology.
So now we will proceed to recover. We are done with the course?
I think that's everything.
Yes. We will then proceed to recover the push cores. This course has they have a small membrane at the top. When you put them in the sediment, the water is expelled through this membrane, but now when you push them out, the membrane is closing and creating a small vacuum holding the sediment inside, and then we put them in a small container and they are safe there. The umbilical, so the cable connecting the ROV with the vessel, which we call the umbilical cord, has, in addition to the fiber optics cables, it has also a copper cable for transmitting energy, and the fiber optics is for transmitting the data. And of course it has also a steel cable to hold the vessel, to hold the ROV and support the strength.
Core number 18 for biology. We started pulling the cores almost immediately. Did we get a shot of how they're all laid out?
Okay. We get the. Did you hear? We get the shot of the push core layout. Yes.
Excellent. Thank you.
We are getting still photos every 10 seconds, but sometimes if we want to get a specific view or
Core number 19, biology.
Here we see holothurian swimming. Or Peniagone maybe, it's the genus of that one. There are some little mysids, krill, swimming in front of the camera. So let's see what has been accomplished. Rocks, Scotoplanus. I see that they collected at least 2 rocks, but I don't see any. 1 bag. I think they collected also 1 bag.
Core number 20, biology. And I think we've got Teresa's holothurians.
Yeah.
And after we finish collecting these, I think we've got a request for a transect between waypoint 12 and 13.
Okay.
So we'll be flying along less than a meter off the bottom, just laser scales on, and at about 0.2, 0.3 meters a second, and that will just give us a transect proportional distribution of the animals that we're seeing. We'll try not to get too distracted, get too excited by seeing cool things. We will of course stop if it's really cool, but we'll try and make some progress. I was chatting to the people who help to drive and decide what the Mars rovers do, and it's quite a diplomatic process. So the scientists make requests and the engineers review what is feasible, and they have a concept called the world's biggest ball of yarn, which is a reference to those sort of tourist traps you get on long road trips where they try and tempt you off your journey with some weird tourist trap. And when they're trying to make progress, when they're trying to do a transect or get to the next waypoint, and all the scientists keep getting distracted by exciting things, yeah, that's what they're referring to. There's a lot of parallels between space exploration and deep sea exploration, and actually the 2 communities have started working together more, both on a technological standpoint, how they solve certain problems and how they organize their science, but also because there may be an overlap. The best potential for life in our solar system right now is actually the ice moons around the gas giants, around sort of Saturn, Jupiter and even Uranus. And those have very similar conditions to our deep ocean here, to the point that if you took an amphipod from one of our deep ocean trenches and transferred it to one of these deep ocean moons, it might actually live long enough to starve. So they can just live in what we hypothesize the conditions are like. There's interesting chemical properties that might be generating free oxygen, and the push-pull, the kneading by gravity of this huge gas giant actually generates heat within the core, and so that might lead to hydrothermal vents or something similar at the bottom of these ice moons. So we're working quite closely with sort of space exploration and deep sea exploration, because we've got a lot of parallels. We realize we face a lot of the same problems.
Yeah, I think it will be very exciting to get in one of these oceans, remote moons. The challenge will be, of course, to keep communication. It's the same problem we have here on the Earth. We are able to communicate to audio worldwide and streaming, because we are connected to a vessel that is connected to a satellite system. But
that's core number 16 and collected.
Use into these oceans. The problem will be to keep communications with them, so there must be probably also a station on that planet keeping the communication with the underwater vehicles and those then sending the communication to Earth. It's challenging.
Until we get quantum entanglement.
Yeah.
I think the delay in communicating with the rovers on Mars is 7.5, almost 8 minutes. So it's not a one-to-one drive. It's a bit more like programming. The commands are calculated and sent, and then executed with a big time lag.
The information that we can get on Earth from satellites is very much restricted to the ocean surface, because we can measure the color, there are also radar signals, but this cannot penetrate the water. Only the few meters in the water surface, in the surface of the ocean. So we have information about the chlorophyll at the surface, the temperature, different biochemical parameters. But with satellites, we cannot get any information from the deep areas of the ocean, from the deep sea, from the deep waters.
Core number 14, biology.
That's the reason that we need to dive with this kind of vehicles that we are using today, in order to get images, in order to get sensors in the water, to get measurements of temperature, salinity, whatever other parameters. We have on board another type of vehicle which is autonomous. It's what we call a glider system. This is measuring the oceanographic parameters like temperature, salinity, chlorophyll, pH, and it can be programmed to fulfill a mission, and it starts then gliding by itself, going to a certain depth by changing the density of a chamber that is inside the glider. And then when it's reaching that depth, it's then going up back to the surface, connecting to a satellite, sending us the coordinates, the data that was recorded, and waiting for more instructions. We have deployed 2 of these gliders here, and they are fulfilling their missions very good. The risk is that one of these may end up under the ice, and then it will not be able to communicate through satellite and may be lost.
I think those are out of the University of East Anglia, I think. And I think you can track them on the website. So you can see what the gliders are up to as well.
Well, yeah, exactly. So the
Core 13, biology.
The mission control of these gliders is in UK. So we just deploy them here, and we can track them here, and also collect them back, bring them to the vessel again. But there are scientists in UK which are controlling these gliders remotely through satellite.
As we have more connectivity, it's amazing the sort of remote work we're able to do. The fact that we're in Antarctica, in a place that's completely underexplored, and able to stream it to folks at home is pretty amazing. I can remember, it wasn't that long ago, I'm not that old, I'm fairly old. My first times out at sea, it was I'm going for about 6 weeks. I'll talk to you when I get back, or maybe I'd get one email out a week, and it would have to be limited text as well. It would be like a telegram. It would be sort of just the facts, really. You know, alive, missing you, cold. And now we can have video calls on board, and we can even share the work with you directly. And it's living memory. It's not been long at all. It's just totally changed.
The sediments here are very, very fine sediments. These are clay minerals, mostly of marine origin. So all the marines know the organisms that are living in the water column. They die, they come to the bottom. This material is processed by other organisms. But of course, we have also some shells and some skeletons from foraminifera and other organisms in the sediments.
12 biology.
And when we have also drop stones that are carried by the continental ice, by the glaciers, and when they break up into icebergs, the iceberg can transport these drop stones, and when they melt, then they leave the stones, drop them to the bottom. So we have seen a lot of drop stones. And there are also some basaltic outcrops that here nearby there's one. When we start the transect, we will see that there is some hard substrate.
Number 11, biology.
So, 2 more to go. Oh, 3. Oh no, there are more.
Oh yeah.
Oh my
yeah.
God.
Well, still got a while to go.
1, 2, 3, 4, 5, 6, 7 to go.
Everyone wants the mud. There's so much fun that can be had. It's maybe we'll try and get some images of the processing that happens once this comes up on deck. It's all fun and games when you're collecting this stuff, but then you've got to process it. So there's a hive of activity in the ROV hangar. We have core extruders and core cutters, and lots of people getting a lot of different information from these. We think that this sediment has probably built up over a real long period of time. It's not been falling very quickly, and so these deep sediments have probably built up over a huge amount of time, and they contain within them a lot of history. Core 10, biology.
Now we see that we have a little bit more current. It's my feeling.
Yeah, glad of a bit of current. The dive we did last night was really, really still, and just couldn't touch the bottom without creating a big cloud that would then hang around for ages. But there was some amazing collecting going on last night, particularly the fish. There was a Chinnichthidae, an ice fish that was collected with the suction sampler without skipping a beat. The ROV just was moving forward, lined up the suction sampler, and slurped it up before it even knew what was going on. And there was a large macrurid, probably a big-eyed grenadier, that was caught with the net. And that's a big one. That's maybe a 60 centimeter long fish. So there was some excellent creature grabbing last night, despite tricky conditions.
We are very close to the
Core 9, biology.
ice shelf, continental border, to the ice border, and there is a lot of icebergs at the surface. So of course there is a constant communication between the ROV control room and the captain of the vessel, because we need to constantly move and also keep an eye that the icebergs are not getting too close to the vessel or to the umbilical of the ROV.
I'm gonna ram home.
Today it was a little bit cloudy. I think we're expecting some snow coming down. And the water here is very cold. It's 0.7 degrees. Normally we have had 1.2, 1.5 degrees water temperature. Or maybe a foraminifera from the water or an algae. And then the ice crystals start to grow, and then of course they float to the surface of the waters where they meet with other ice crystals and start building what we call pancake ice, which during the winter will grow and grow until 1-2 meters magnitude. And this is the marine ice. In addition, we have the icebergs, where they are coming from the continent. This is freshwater ice. This is compacted snow that is falling in Antarctica for thousands and thousands of years. And of course, this ice is not static, but it's also moving slowly, like in glaciers. And when it reaches the border in the ocean, it can break, and then we have the icebergs floating on the ocean.
Got a question in the chat of what's the most memorable creature sighting. Do you have one that springs to mind, Pedro?
Here in this cruise, what I find very, very beautiful in these corals that we are finding on the hard substrate. We have visited some outcrops that were really vertical walls of rocks, and they were completely covered by beautiful, colorful corals and sponges.
The geologist got a bit extra with this one. It wasn't this cruise, but it was another Schmidt Ocean Institute cruise. The one that jumps out to me as the most amazing observation was in 2014, and the original Falcor. I was out with our team in the Mariana Trench doing some very deep lander work, and we were sort of pushing the limits of how deep we thought fish could go. We'd heard a rumor that there was a snailfish very deep in there, but we hadn't had sort of confirmation of that. And we'd found the snailfish, we'd seen them, and that was all sort of very exciting. And it was 3 a.m. and there was only 3 of us awake. There was myself, Alan Jameson, my PhD mentor at the time, and one of the engineers, and they were just looking back over a task camera that was actually just checking that the corer had gone in. So all it was meant to do is just show that these cores had gone properly into the seabed. So we weren't expecting anything exciting going on. And the engineer just leans back and says, like, "Oh, you guys like fish, check this thing out, it's pretty cool." And just deeper than we thought fish could go, an incredibly beautiful animal swam into shot. Just did anyone catch the call number on that one? It was a bit muddy.
That was 5.
It was 5. Okay, that's call 5 for geology.
Look at polychaete moving in the water. Yes, go. It's curious to see what we are doing.
We're probably stirring up some good food.
That's a planktonic species. It's a Tomeropsis.
So that fish that we caught on the video, deeper than we expected, that is what we then went on to call the ethereal snailfish, and it features in Blue Planet too. If you remember that one, it's that beautiful, long flowing fins. It looks like the Ghost of Christmas Past, and it was just incredible. It took us totally by surprise. We were running around, banging on people's cabin doors, waking them up, and just amazing. And that thing still holds in my mind. It took so much technology, it took so much brute force for our equipment to go to over 8,000 meters deep, and then this very fragile, very beautiful animal was living there and just harmonizing with that pressure, and just really drove home sort of how separate that world was. That's number 6, geology.
The density of the water is controlled by the temperature and the salinity. More saline water is more dense. So fresh water by itself will be on top of marine water or saline water. But there is a combination of both. Of course, if you have warm saline water, this may be less dense depending on the salinity than cold fresh water. The density anomaly of the water is at 4 degrees. This is the more dense water is 4 degrees. Less than this, it gets less dense. But this applies, of course, to the still water, to poor water. If you have more salinity, then this 4 degrees barrier gets lower, so you can have more dense waters at 1 degree if you have you have marine salinity. And when you have very, very cold water and very, very saline water, a lot of salts, then it gets very dense and gets trapped at the bottom. It cannot move from there because the density is so high. So if you have little depressions, that areas that are a little bit deeper, and you have these conditions, then it may be that some of the water get trapped on the bottom and is not contributing to the water circulation then.
Long course 7, geology.
I would let that clear. I would take this one out, put it at the time. Or it's up to you. Yeah.
Yeah, exactly. This will be these brine pools. They are in different oceans, also in the polar regions. And the problem when the water is not circulating, when the water gets trapped in some areas, is then the biology starts consuming the oxygen that is in the water. And I mean, it's not a problem, it's actually a condition. And then these brine pools can also get less oxygenated or even anoxic. It's a paradise then for different type of microbes that they are living in these extreme situations, like extreme pressure, cold, salinity, lack of oxygen. Normally animals cannot live under these conditions, but there are a lot of microorganisms that they can.
Slowly. Just as you bend a little bit. And they go, they just there. Let it adjust. See where it goes.
Having a close look at this manip, you can see our little coral snipper, that red block on the other, on one side, and then meets with a little bit of a blade on the other, and that lets us take snips out of corals and sponges and things like that, without having to sort of tear or do more damage to the organism. A lot of these things aren't particularly strongly rooted, and so you can damage a very old colony and take more than you need. And so it's nice to be able to just take a little snip and leave the colony intact. That is core number 3, long core for geology. I think we're down to our last one, core number 8.
Fiona? Can you? Creator is coming to visit us and see what we are doing here.
Yeah, we've gathered quite a following, staying still with our lights on. Seems to be a lot more stuff in the water column now. The deeper dives earlier was loads and loads of marine snow, but there wasn't much swimming past. The odd comb jelly. But it's busy in the water column now. Okay, core number 8, geology. That's the full set. Well done, everyone.
Great.
Okay, we'll gather our composure and get all our toys in, and then we're on to a transect.
you see in the water, there's a small mysid shrimps. This is also what we call krill, Antarctic krill. And they came attracted by the light of the ROV. We are not so deep, so 350 meters. We have not seen so many of them in deeper waters, but here they are more abundant. Now we are done with the bush coring, and we are going to proceed with the visual video transect.
We're going to try not to get too distracted as we do a video transect to the next waypoint. So in order to do that, we'll fly at just under a meter off the bottom. We'll move at about 0.2, 0.3 meters per second. Is it meters per second? Per minute? Yeah, it wouldn't be that fast. And we've got our lasers on, which give us a scale. So those two green dots you can see in the center of the screen, they're 10 centimeters apart. And so when we have the speed of the ROV, we have a scale. We can then work out the density, sizes, and even the biomass of these animals. Allows us to characterize the habitat.
So here we go. There are plenty of holothurians, sea pigs or sea cucumbers. We have different species. This is my favorite. And there's some sea stars, or brittle stars, better. Also very little small holothurians, probably. And here we are reaching a hard substrate outcrop. Here you see also some corals, some sponges.
Well, this is quite an abrupt wall.
Sea urchin. No, there's a wall in front of us. We have the radar sonar system telling us that the wall is quite massive. There are some crinoids on the wall and some corals.
I think that was it. I think it was just a big step.
Yeah, a step. And we are now at 350 meters and climbing up.
So we're just leaving waypoint 12, heading towards waypoint 13.
There's a small fish over there. The density of the sea cucumbers increased a lot. Some little sponges.
We're transecting, but I've just got a request that if we do see coral on the rock outcrops, to take a sample. This looks like the apex of this area. Okay, here we go.
Here we see some brittle stars, sea urchins also. Now we see the map with our waypoints. Before we start diving, we produce an own map of the whole area because the maps, actually there were no detailed maps from this area. Specifically for this area because it was covered by ice, but also from other areas that we are visiting here in our Antarctic cruise. We need first to produce our own detailed maps in order to plan the dives. very bumpy here. Lots of small hills and valleys. And lots of holothurians, these little creatures that we see at the bottom. They are dominating this community. In more depth, in 600 meters depth, a little bit more, we had a lot of sea urchins, the little reddish ones that were dominating that community. Also, we had a lot of worms, nemertini, ribbon worms, very large ones, 1/2 meter long.
Something's crashed down here.
Yes. What could this be?
That could be, how wide is it? It's quite wide. It could be whale feeding. Maybe even seals.
Yeah,
we're shallow enough now.
That's true.
That they're rooting around in the sediment. That's another one of those.
Ah, that's a wonderful one. Yes, beautiful.
We started our shift with a beautiful shot of one of those. Undulating terrain here. We're sort of hopping over these little mounds. Sometimes they're still sediment covered, sometimes the rock is poking out. We were surprised last night how cohesive, how sticky the sediment is. Even quite steep slopes, it seems to be holding onto the rock. Usually I expect them to be bare. So this is very fine sediment that's built up over a long amount of time. It's quite sticky, and I think maybe the current speeds are generally quite low here.
And we saw another of these big polyps. They seem to be quite common here. We have not seen them in deeper areas. We see that they are small and larger of these holothurians. I believe that these are juvenile, so we are seeing here probably a reproduction event.
Wow. The gathering.
Yes. So they are probably juveniles from this year, that they are still growing, and then we have probably grown up ones from last year.
We're flying quite high. I think these are almost 10 centimeters.
So the laser pointers, these are these green pixels in the middle of this screen. The distance between them is 10 centimeters. This is helping us to get the impression.
Just do a little bit of ice avoidance for the umbilical. Get back underway to the waypoint now.
Okay, we have some mention from the captain that there is some ice on our way.
There is so many here, and it's all the same type, isn't it?
Yeah, it seems to be same type.
Oh, we've got one sea pig in there. Went to the wrong address.
Yeah.
Really abundant. So obviously this sort of tabletop peak is collecting plenty of sediment for them.
We see another rock. Thousands of holothurians.
Really is. Somebody's going to have fun counting these. But with a transect like this, known vehicle speed, scale bar in view, we'll be able to work out the density of these animals. It's certainly different from the other sites. This is the holothurian party.
There's a question in the chat if these are squids. No, these are sea cucumbers or holothurians or sea pigs are also called. We have seen some squid, but they are not so common. Tom, I think this is a question for you. If there are some squid down here.
Yeah, we've had a few. We've had a few visits from the glacial squid. I've got a low light bioluminescent lure camera that I've been lowering into the water, and we've had a few interesting visits on that. So the squid do seem to like it. They seem to come in pretty hot actually. We've had a few of them crash into it. There's quite a few squid in the area, including the famous colossal squid.
We see also here some chunks of sediment. I don't know where they come from. And a brittle star who lost one of the arms. There's some velula corals. What happened here?
Yeah.
Can we take a picture of this?
Which one?
The hole.
Wow.
Maybe a drop stone that went right into the sediment?
So I think this may be also gravity coral or something like that.
You think it might be us? It's quite round.
I don't know.
Oh, there's something interesting here. See the isopod?
Where is that?
Just.
Can you get me that one?
Yes, I will.
It's not the one you wanted, but it's close enough.
So just up and to the left there is a large pillbug looking thing.
Which one?
This little guy here. The one that looks like a trilobite. We can zoom and get some shots before we grab him.
Oh yeah, that's the cerulid isopod. Cerulid is a family that is very common here in the Antarctica, and then they have some representatives also in the deep sea, but I think only, I'm not sure, most of them in the southern hemisphere. I think there are a few of them in the northern hemisphere. That's a brittle star, and now we are going to try to collect one crustacean, one isopod. They look like trilobites from the morphology, but of course they are not related to trilobites.
You reckon slurp gun?
Yeah, here it is.
Nice.
Yeah, slurp gun, yeah, that's a possibility.
I'm gonna crush it. Oh, that's a great animal. Yeah, it's totally a trilobite.
Yeah, here we go. Excellent.
Just they look very, very similar.
Excellent.
Bit of convergent evolution. Trilobites are quite a separate group, but I just can't help but think that whenever I see these. Very, very cool. Do you reckon slurp?
Yeah. So we didn't
Crush it?
You're going to slurp it?
Yep, let's go for it.
Where's that? It's from the sun. Oh, it's close.
Oh, the fish came up to watch what we are doing here.
Sample.
Great, ROV, please come to stop. We're gonna be doing taking some samples here.
Okay, come to stop.
Thank you.
Please keep your eye on the eyes and Tom.
Hopefully this fish doesn't eat it.
Sometimes we're busy and trying to get position.
So now we need to stabilize the ROV. So one of the legs will be holding us at the bottom. And then we are going to use the slurp gun to collect this isopod, this crustacean.
Okay, where do we want to put this?
We're pretty open. What are we set to right now? Is that 3?
That's 3.
Yep, 3 looks to be clear.
3? Okay. Stand by. I think that will hold. Okay. Yeah. I can't see it on my screen. Where's it on my screen?
It shouldn't be able to move quickly. It won't do like
Oh, there it is.
a prawn
It's right in
pink.
front of the nozzle. Is that in front of the nozzle? Yeah, got it.
Nice.
Okay.
Oh,
Here we go. Now it's
running, running.
trying to escape.
There you go, got it.
Yeah.
Increasing suction. It's kind of want to.
It's still there. It's still there.
It's still where? In the nozzle?
Yeah, see?
Oh, okay, increasing nozzle. Increasing suction, full suction. Checking.
Oh yeah. Oh, is he too wide? Might be too big.
Oh, we could dump him in a bio box.
Yeah, now that we've caught him gently. It's a great
Yeah.
shot of the underside. Okay, let's transfer to a buy box. So-
Slow down suction immediately, right?
Yeah.
Okay, so we better do fast on this one.
All of BB4 is empty.
Watching out. So I'm gonna keep an eye on that before it runs off. Okay. E4. I'm going to turn off suction, watch it. Suction's off.
You can just see the edge of him there.
If you don't mind, watch that tongue trip if he falls
Okay.
in.
Okay.
You have better eyes than I do.
Here it goes.
Shake, shake, shake.
See. Yay! Well done.
B4, closing B4.
BB4B isopod.
Must be the too rich. Sorry, that was just too much for me. They're approaching him.
Anything else we do like that?
Do you see any corals on the rock?
I think there will be some. Let's take a look. That way here, maybe we can just look with the camera.
Something quite mat-like on the left.
There are some
On the right,
bryozoans, shrimps. We see shrimps. A lot of shrimps.
I think that's an old sponge that's broken away.
Old sponge. We have some serpulid worms.
And
We have some sea urchins, but I don't see any coral.
How about that one?
Those are bryozoans, but they do a good job tricking us.
Yep, here we go. No,
No,
serpulid worms. Shrimps.
Alright.
I think we...
All of those.
Those all bryozoans, Pedro?
Yeah.
Even that one on top, the branching?
Nothing interesting?
No corals.
No, okay.
Should move.
No, I can approach out. You want me to ram out? Start with ram. Coming out. Wait. You got it?
Well done on that one. That was great. Okay, well, we're in a good spot. We're just going to swap out some of the pilots so that everyone can get dinner. And because this takes quite a lot of focus, this exhausts you quite quickly.
Shall we go?
Those people joining us on the stream, we've reached the time limit for this particular stream. So what we're going to do is we're going to bring this one to a close, and then we're going to join you again on a part 2. So it's not the end of the dive, you can continue to watch, but we just have to jump over to a fresh stream. carry on. Okay. So we may as well keep the scoop in hand while we look for the third.
Change file now, so send them up using a blockchain. Think about it.
So,
Anyone lost us just for a while there? We are changing the streams, so you might have heard us muted for a while. I think there's a limit to how long we can stream on YouTube.
so we came to an end of our shift. Now, new scientists will come and stay with you. Thank you very much for listening to us. It was quite fun to take all these deep sea samples and show you the Antarctic deep sea. Thank you to the ROV pilots also for the excellent work. See you another day.
Hear a bit of noise now just as we're changing over. It's the dinner rush. So we're changing both the pilot team and the science team. But for everyone who stayed with us for our shift the last couple of hours, it's lovely to have you along. Really good questions.
Big one. Big pig. All right. Round ones.
And for this.
Round and pig.
So how are we?
Big leg.
So we're just looking for one last sea pig. So we've still got the scoop in hand. One of the fat boys.
What's here? I imagine.
There. Yeah.
Okay.
And there. All the cores done.
Both papers are pretty free. We have 3 colors. On the backs.
I didn't take any, but
Okay,
looks
so we
like
can take one.
one statement, but there's still two
Okay.
to play with.
Oh, really? Oh, this is fun.
Go
Can you
nuts.
imagine that?
Go nuts.
Let's see.
We choked on an isopod and then spat it out and put it on the buy boxes, so we're
Is the
pretty...
one the tribal bike?
Yes, yeah, I like it.
Yeah.
Thank you, gang.
No, thank you.
Thank you.
See you later. You found them? Oh, there is.
They're shuffling papers.
I was doing my laundry actually. See? Until now. This is done. Yeah.
Yeah, that's because you didn't reuse the same set.
Oh.
So probably the.
Can we have a quick zoom in before we pick it? No? Otherwise, no? Okay.
Yeah, we'll get set first.
Alrighty.
Alrighty.
We're just gonna redo it, I guess.
There.
Thank you. Yes.
Pretty soon.
Oh. And there's a tiny brachiopods in the bottom. Yeah.
Thank you.
Thank you so much.
From these fish in the oceans, I love that.
Yeah.
I'm just leaning on stuff.
What?
Come on, fish, run away. We don't want you.
Fish and everything.
Yeah, you see, it doesn't move. Oh, come on. Goes into BB4A.
With the other two?
Yes, with the other two.